Okay. Hi everyone. Good evening, guys. Welcome back to the channel. Good evening, all of you. All audible and visible. Please let me know. This session is going to be huge and it's going to be a long session, a marathon session where we are going to complete our class 11 NCERT with highlights of PYQs. Good evening, all of you. Good evening. How are you all? Let me know in the chat section how excited were you for this session. How excited? And quickly like, subscribe, and share this session to all. And also, I thought I should complete this NCERT today. One, because from Sunday onwards, guys, that is tomorrow, 19th of April, we are going to have mock test series, free mock test series starting from tomorrow from 2 p.m. to 5 p.m. So I wanted to complete this at least class 11th NCERT today, and tomorrow at the same time we are going to complete with our class 12 NCERT. Good evening, all of you. Eagerly waiting. Quickly share this session with all your friends, and we are going to start with it quickly. Give me a thumbs up, and please get ready with your NCERTs. Those of you who don't have your NCERTs in front of you, not the right way. Today we need NCERTs and a highlighter. Good evening. All right, guys. Is it really useful for freshers? Yes, why not, Siva? You'll get an idea. And I am going to underline only those lines which are very important, from which we have got the PYQs. Okay? So quickly, all NCERT is ready, and highlighter. Good evening. Pencil. Okay, whatever. But if you have not marked anything, only then the pencil would work. Okay? 360 possible, absolutely possible. See, these are the key words that we're going to study today. New NCERT or old. I am going to use new NCERT today because certain changes have been made in the new NCERT. So let's use new NCERT. The old NCERTs will be covered in your unit-wise revision. Those topics will be taken care of. But today we are going to read through the new NCERT. Okay? All right. Session timings, we are starting now, 7:30. And I don't know. Approximately it will go till 11. So let's give a quick start. Okay. We'll talk about that, Jitu. Not the right time. I think the best way to handle your anxiety and panic attacks before your preparation is to revise things. You know, when I have always believed that knowledge is followed by confidence. Confidence is not some sparrow which will just come and sit on your shoulder. It comes with your knowledge, right? Once we are well prepared for the exam, there is nothing which can shake us. Okay, how many chapters are going to cover? We are going to complete the complete NCERT. Now, no more. Yes. Let's start the NCERT. I'm going to go backwards, okay, starting with human physiology. So, starting with chapter number 19, give me a thumbs up that you're all set on chapter number 19. Yes, all chapters will be completed today. And starting now. Okay, the first line is the neural coordination is fast but short-lived as the nerve fibers do not innervate all the cells of the body. So this is assertion and reason. Then talking about endocrine glands, they are ductless glands, secrete hormones released in the blood. A distinctly located target organ can be reached because blood is going to carry the hormones. Now, what is the definition? It is a non-nutrient chemical which acts as an intercellular messenger and are produced in the trace amounts. Moving on to the next. Now see the diagram also. All the labelings should be very clear. Don't miss any diagram. Okay. Then coming on to hypothalamus. What is hypothalamus? Where is it present? The basal part of diencephalon, which is a part of forebrain. Now it contains several group of neurosecretory cells which are known as nuclei, and these produce hormones. Now these hormones are going to regulate the secretion of pituitary hormones. Now these hormones can be of two types: releasing hormones and inhibitory hormones. Releasing are going to excite or stimulate the pituitary hormones, and inhibitory are going to inhibit the pituitary hormones. Is this clear? Then there is one hormone known as growth hormone, inhibitory hormone, also known by the name of somatostatin. So this inhibits the release of growth hormone. Are these all points clear? Are these all points clear? Sequence will be from backwards. Okay. Next, moving on. So pituitary gland. Coming on to pituitary gland. So where is it located? Pituitary gland is located in the bony cavity called sella turcica. It is attached to the hypothalamus by a stalk. Okay. And anatomically, pituitary gland has two parts: adenohypophysis and neurohypophysis. Can we see that? So the two parts are adenohypophysis and neurohypophysis. Adenohypophysis is further differentiated into two regions. One is pars distalis and the second is pars intermedia. Distalis, which is known as anterior pituitary, it produces these hormones: growth hormone, prolactin, thyroid stimulating hormone, ACTH, okay, FSH, and LH. While pars intermedia, which is also a part of pars distalis, adenohypophysis. Now, pars intermedia secretes one hormone known as MSH, which is melanocyte stimulating hormone. Now, pars intermedia is not very distinctly seen. It is almost merged with pars distalis. Are these points clear? Everyone is able to underline and revise these important lines. Then, neurohypophysis, also known as posterior pituitary, it doesn't produce it doesn't produce any hormone. It only stores and releases the two hormones oxytocin and vasopressin, which are actually synthesized by hypothalamus. And how does it transport? Hypothalamus is going to transport these two hormones axonally. That is, the axon endings are going to open into the posterior pituitary to neurohypophysis. All these points clear? Clear? Perfect. Then over secretion of growth hormone leads to gigantism, while the low secretion leads to pituitary dwarfism. Now, if this higher secretion, hyper secretion or over secretion of growth hormone occurs in the middle age, then it leads to the condition acromegaly, which leads to serious complications or even premature death if unchecked. Are these points clear? Clear? Moving ahead. Then prolactin, which is secreted by anterior pituitary. It helps in the growth of mammary glands and formation of milk. Then moving on to gonadotropins. Two types, LH and FSH. LH stimulates the androgens from, that means the secretion of testosterone from testes, and FSH and androgens together they regulate spermatogenesis in males, while in females LH induces ovulation. Now, FSH in case of females, it induces or stimulates the growth and development of the ovarian follicles in females. Are these points clear? Everyone clear? Okay, MSH, that is melanocyte stimulating hormone secreted by the pars intermedia. It regulates the pigmentation of the skin. That is why the name is MSH. Melanocyte means melanin producing cells are stimulated. Oxytocin is known as a childbirth hormone or milk ejection hormone because of the functions it performs. Vasopressin is going to help in reabsorption of water and electrolytes. Is this point clear? Aditya, we can't go slow. We are going to quickly revise everything. You remember everything. Guys, please. Now, I'll tell you one thing, small thing in between. See, many of us, we are going to feel as if, we are going to forget everything. No, just stay calm, go quick, okay, and revise everything. Go very quick and revise everything. All chapters will you do? All chapters, all chapters. Now, it reduces the loss of water. What does vasopressin do? It reabsorbs water and electrolytes from the DCT and CT collecting tubules and reduces the loss of water. So, urination is decreased. That is why it is known as anti-diuretic hormone. Is it clear? If ADH is lesser, is produced in lower amount, then it leads to the condition known as diabetes insipidus. Is this point clear? Which will lead to excessive water loss and dehydration. Pineal gland, dorsal side of the forebrain. This is where it is present on the dorsal side, and it releases a hormone known as melatonin, responsible for 24-hour diurnal rhythm, sleep-wake cycle, body temperature, and influences the metabolism, pigmentation, menstrual cycle, as well as the defense capacity. Moving on to thyroid gland. Now, thyroid gland, thin flap of connective tissue called isthmus is present. Thyroid gland has two types of things, follicles and the stromal tissue. Follicles have follicular cells which secrete two types of hormones, tetraiodothyronin and which is T4 and triiodothyronin, that is T3. Tetra because it has 4 atoms of iodine, tri because it has 3 atoms of iodine. Iodine is essential for the production. If there is low amount of iodine in your blood, thyroxine will not be produced and it will lead to the condition known as hypothyroidism. Is this clear? And it leads to enlargement of the thyroid gland. The condition is known as goiter. If hypothyroidism occurs during pregnancy, then there will be defective development and maturation of the growing baby. Condition is known as cretinism. It will lead to mental retardation, low intelligent quotient and abnormal skin. And otherwise, in females, low thyroxine can lead to abnormal or irregular menstrual cycle. Now, cancer of thyroid gland will lead to hyperthyroidism, which is known as exophthalmic goiter. Exophthalmic, why? Because the eyeballs, X means out, ophtha means balls. So there will be protrusion of eyeballs. This is also known as Graves' disease. Are these points clear to everyone? Are these unit-wise revision will go on, Shri. Don't worry about that. Please concentrate here. We are just going to read NCERT. Unit-wise revision will continue from Monday onwards. Okay. So, thyroid hormones control the metabolism of carbohydrates, proteins, and fats, and maintenance of water and electrolyte balance is also affected. Now, there is another hormone produced by the thyroid gland, which is called thyroid gland. Sorry, TCT, thyrocalcitonin. Now, this is going to lower the blood calcium, so we call it hypocalcemic hormone. Then there is another gland, parathyroid, present 4 in number, present on the backside of the thyroid gland, secretes a hormone, parathormone, which increases the calcium. So basically TCT and PTH are antagonistic to each other. In order to increase the calcium, bone resorption takes place, resorption of calcium from renal tubules, and increases the calcium absorption from the digested food. So since it increases the calcium, we call it a hypercalcemic hormone. Are these points clear? Then moving on to thymus. So thymus is a lobular structure between lungs and sternum on the ventral side of aorta. So note down these locations very carefully. It produces thymosin, the name of the hormone, and these thymosins are important for the differentiation of T lymphocytes. Is that clear? Now T lymphocytes, we know that they are responsible for cell mediated immunity, and they're going to stimulate the production of antibodies to provide the humoral immunity. Now, in old individuals, since thymus gland is degenerated, which is responsible for decreased production of thymosin and hence weak immunity. Are these points clear to everyone? All clear? Quickly let me know. Adrenal gland one. Okay, so adrenal gland is responsible. So it is present one on each kidney. First of all, it is present one on each kidney. Now there are two regions. So outer region is cortex, adrenal cortex, which produces corticoids, and the underproduction of these corticoids alters the carbohydrate metabolism, causing acute weakness and fatigue, leading to Addison's disease. So remember, Addison's disease is due to underproduction of all the corticoids. Are these points clear? Right? Moving ahead, then we have adrenal medulla, the inner part of the medulla, which secretes again two types of hormones, adrenaline and noradrenaline. The same hormone which has increased in surplus in your condition these days. Are these points clear? Then, together, these two hormones, adrenaline and noradrenaline, are known as catecholamines. These are emergency hormones, response to stress during the emergency. That is why we call them hormones of fight and flight. What do they cause? See, these days, since you are in adrenaline rush, your alertness has increased, your speed has increased, your pupillary dilation, you're more vigilant. There is piloerection, means the hair present on your skin, they are in the raised state. Then sweating, heartbeat, strength of the heart contraction, rate of respiration all increase. Now since it is an emergency condition, so it stimulates the breakdown of glycogen to produce more energy. Also it leads to breakdown of lipids and proteins. Now cortex is further divided into three regions. The inner one is reticularis, middle one is fasciculata, and the outer one is known as glomerulosa. So GFR, from outside to inside it is GFR. We remember GFR, glomerular filtration rate. So use the same mnemonic here. GFR is glomerulosa, fasciculata, and the inner one is reticularis. And all these hormones are collectively known as corticoids. Now see, all the three layers, they produce different type of corticoids. Now, one is glucocorticoid. Gluco, as the name says, responsible for carbohydrate metabolism. Second is cortisol. Cortisol is a mineralocorticoid. One example of glucocorticoid is a cortisol. Okay. Now, then there is second, which is mineralocorticoid, and very important mineralocorticoid is aldosterone. As the name says, it will maintain or regulate the balance of water and sodium electrolytes. And then these glucocorticoids, see, stimulate gluconeogenesis, new glucose formation, lipolysis, so fats are dissolved, proteolysis, and they inhibit, wait, and they inhibit, just a second, guys. Yes, yes, yes. Just a second. Yes, 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 yes. Hello, hello. Just a second. Hello, hello, hello. Yes, please. I know that, but it was already discussed that 1 hour we can go in parallel. Cool. Okay. Okay, okay, okay. Mm, all right, guys. Welcome back. Yes. Okay. Moving ahead, moving ahead. I thought there was some tech issue, but no, everything is fine. Just a second. So where were we? We were already on thyroid gland. We were already on thyroid gland. Wait. Guys, when the time is less, you actually have to be very, very particular with the time and be sure. Okay, now you have to restrict yourself that, okay, only this much time for this chapter. Okay, that's what we're going to do today as well. Now, talking about glucocorticoids, they stimulate gluconeogenesis, lipolysis, and proteolysis and inhibit the cellular uptake and utilization of amino acids. Okay, then glucocorticoids, particularly cortisol, it is anti-inflammatory and suppresses the immune system, and cortisol is your stress hormone. That is why during your stress your immunity goes weak. Then okay, cortisol, but it stimulates RBC production. Aldosterone is going to stimulate the reabsorption of sodium and water and excretion of potassium and phosphate ions. While there are third type of corticoids also, which are androgenic in nature, and they're responsible for the growth of axial hair, pubic hair, and facial hair during the puberty. Are these points clear? Pancreas, a composite gland, or you can call it a heterocrine gland, and the endocrine part we are going to discuss here consists of aisles of Langerhans, 1 to 2 million cells. 1 to 2 million. These cells are known as aisles of Langerhans and they constitute 1 to 2% of pancreatic tissue. Out of the total size of pancreas, only 1 to 2% is endocrine in nature. There are two main type of cells. See, there are others also, but the two main types of cells present in aisles are alpha cells which produce glucagon and beta cells which produce insulin. Glucagon is going to increase the blood sugar level. That is why it is known as hyperglycemic. Okay, so what is the target? Remember, it acts on liver cells, that is hepatocytes, and stimulate glycogenolysis. So breakdown of glycogen occurs, and glucagon reduces the cellular uptake so that high level of sugar is maintained in your blood. While insulin, the target organ is hepatocytes and adipocytes, the fat tissue, right? It enhances the cell glucose uptake by your cells so that the blood sugar level decreases. So remember, again, glucagon and insulin are antagonistic. Insulin is going to lower the blood sugar and glucagon is going to increase the blood sugar. All clear? Then hypoglycemia. See, now, this is done. So, blood, since it decreases the blood sugar, we call it hypoglycemic, right? Now, prolonged hyperglycemia, if there is prolonged increase in blood sugar, that condition is known as diabetes mellitus. See, don't get confused. Diabetes insipidus is due to lower secretion of ADH, while diabetes mellitus is due to lower secretion of insulin. Make sure that you write it somewhere. Write it here in the white space, but do write down. Okay, moving on. Wait. Yes. Then, okay. Then. Wait. Yes. So prolonged is known as this diabetes mellitus. Moving on. Now, what is the sign? What is the symptom of hyperglycemia or diabetes mellitus? You see the presence of glucose and ketone bodies. So these only form the test of diabetes mellitus also. So glucose in urine is glucosuria, and ketones in urine is ketonuria. Are these points clear? Moving on to testes. Testes produce androgens and regulate the development and maturation and function of male accessory sex organs. All these these epididymis, these ducts. They stimulate the muscular growth, facial and axillary hair, aggressiveness, low pitch of voice, and regulate spermatogenesis, also responsible for sexual behavior, libido. And they are anabolic. They are anabolic means they help in muscle development. Then talking about ovary secretes estrogens, which is responsible for the growth and activity of secondary sex organs as well, apart from oogenesis. Ovarian follicles, presence of female secondary sex characters, memory development, then sexual behavior again controlled by estrogen in females. Now progesterone is known as a pregnancy maintaining hormone. It acts on the mammary glands, stimulates the formation of alveoli, which stores milk and milk secretion. Now there are other organs also which are responsible for hormone production. For example, the atrial wall of heart secretes your ANF, which decreases blood pressure by dilating the blood vessels. Then there are juxtaglomerular cells of kidney. We'll talk about it in excretion also. They produce erythropoietin, which stimulates RBC production. So JGC is known to produce two types of hormones. One is renin. We'll talk about it later, which increases the blood pressure. Second is erythropoietin. It acts on your bone marrow and stimulates the production of RBC. Clear? Then your GIT, that is gastrointestinal tract, also produces four peptide hormones: gastrin, secretin, and CCK. Gastrin, it is going to enhance the gastric glands to produce the hormone like enzymes like pepsinogen and HCl. And then secretin is going to act on the exocrine part of pancreas to stimulate the secretion of water and bicarbonate, while CCK is cholecystokinin, acts on two target areas, pancreas and gallbladder. So it enhances the secretion of both pancreatic juice to secrete their enzymes, and it will stimulate the liver to produce more of the gallbladder to release bile juice, while GIP inhibits the gastric secretion. Is that clear? Then there are certain non-endocrine tissues which secrete growth factors. Are these points clear to everyone? Please let me know. Are these points clear to everyone? All right. Now, moving on to the mechanism, very important area, very important aspect. See, hormone receptors are present on the target tissues only. So, every hormone has a specific receptor and they will be found on that only area where the hormone should act. So, these hormones will bind with the membrane-bound receptors. These receptors can be present on the membrane, then they'll be known as extracellular or membrane-bound receptors. And if these receptors, hormone receptors, are present within the cell, especially within the nucleus, they are known as intracellular receptors. Is that clear? After binding with the receptor, it forms a hormone receptor complex, and this complex leads to some biochemical changes and eventually the physiological changes. Now, hormones chemically can be divided into four different types, and you must know which hormone belongs to which category. Is that clear? Now, the hormone, see this pathway. Now we are talking about these two. See here. So the peptide hormones and the amino acid derivative hormones, these two hormones will always have membrane bound receptors. Okay. Now membrane bound receptors are so the hormone is going to bind on the plasma membrane, on the membrane. So because the hormone cannot go inside the cell, so there is need for generation of secondary messengers also. Now these secondary messengers could be cyclic AMP, calcium, inositol phosphate, okay, IP3, which we call. So these are the second messengers. So very important to remember that secondary messengers are only needed when the hormone binds to the extracellular or the membrane bound receptor. Are these points clear? While we have another type. So the other two types, one steroid hormones and T3 and T4 iodinated hormones. These two type of hormones have their receptors within the nucleus. Very important to remember. Very important to remember that which T3, T4 and what T3, T4 and steroid hormones, they are present, their receptors are present within the nucleus. Are these points clear? So hormone receptor complex is found here in the nucleus. It will directly lead to some genetic changes, and these changes are going to be almost permanent. Are these points clear? When the topic important and explain in detail. Darling, please listen. We are continuing with the detailed revision. Unit-wise revision will continue. Okay. So right now it's important that we highlight the important lines of NCERT so that every time we revise, every time you come to the class and unit revise revision, your NCERT is already highlighted. And also please remember, from tomorrow onwards your mock test series are starting from 2 to 5 p.m. daily. Very, very important. So that is why I have taken it as a priority to revise the whole NCERT. Okay, now I would suggest you one thing. In case there is some difficulty, if you feel that okay, this particular topic is difficult, you keep writing down that topic, make a list of that topic, so that whenever we come to the unit wise revision class, we are able to focus more on that. Is that clear? Perfect. Yes, unit wise revision will continue, don't worry about that. Okay, so guys, with this the first chapter is over. And now we move on to the next chapter, which is neural control and coordination. Please let me know that these points are clear to all. The first chapter is clear to everyone. Please let me know, give me a thumbs up that first chapter is done and clear to everyone. I am running. Yes, I am running. It's not just you who's in the adrenaline rush, so am I. Now, coming on to the second chapter, that is chapter 18, we are going backwards. Okay, yes, going back to front, back to first. So, first, neural control and coordination. So, body needs to be coordinated to maintain homeostasis. So, why do we need these two chapters? These two systems to maintain homeostasis? So neural system and endocrine system jointly coordinate and integrate all the activities in a very synchronized manner. So see, what is the difference between the two coordinations? The neural coordination is point-to-point connection for quick coordination, while endocrine system works slowly through hormones. But the effect of endocrine system is going to be longer. Is that clear? Are these points clear to all? See, those of you who are present in the class, they are going to be a step ahead. Those who are going to join me in a little while, they will be missing these topics and they'll have to listen it later on. But never mind, never mind. All right, coming on to the neural system. So neural system consists of highly specialized cells called neurons, right? They are known as neurons. Now these neurons were they were not always so elaborate like in our case. In Hydra, in very lower invertebrates, they were of very small size. But in in higher organism like insects, there is a brain present, number of ganglia and neural tissue. And then look at us, we have even more elaborate system. Now in humans, the neural system consists of two components. One is the central nervous system, CNS, and peripheral nervous system. Central nervous system has further two parts, which is brain and spinal cord, while peripheral nervous system consists of the nerve fibers. Now there are two types of nerve fibers, afferent and efferent. Afferent are going to carry message from tissue to central nervous system, and efferent is going to carry message from central nervous system back to the organ. Are these points clear? You missed chemical control, never mind. Never mind, you can see, let's go with the flow. Wherever you join from, go with the flow, and you can always come back and see the recording for that one chapter. Okay? Yes. Never mind, never mind. So you can always join in. All right. Now see, central nervous. So PNS is further divided into two more divisions based on whether the system is going to take care of the voluntary organs or the involuntary part. So in case the central nervous system is somatic neural system. Let's talk about somatic neural system first. So somatic neural system is between central nervous system and skeletal muscles, your voluntary part, right? And autonomic nervous system is going to work between your central nervous system and involuntary organs and your involuntary muscles, smooth muscles. So autonomic nervous system can be further divided into two categories, sympathetic and parasympathetic. Sympathetic is which works during the emergency conditions, right? And parasympathetic is which is going to work during your normal conditions. So for the entire 2 years, you have been using your parasympathetic nervous system, but now at this time, it's the sympathetic nervous system which is already activated. Yes, hard part is to remember to not to study. Hustler. So when you repeat things, you obviously remember. See, we are not some, we are not suffering from any dementia. We are very young for that. So we are all healthy. We are all in the right state. Panic makes you feel that way. Otherwise, we are definitely going to remember everything. Don't worry. You have already highlighted, then make sure that you're revising it with me. And in case you've missed any point, you highlight it. Is that clear? So you don't have to highlight over the highlight, but obvious. All right. Now, visceral nervous system is a part of PNS, which consists of fibers, ganglia, and plexus from CNS to the viscera and from viscera. Viscera means from internal organs. Is that clear? Coming on to neuron. Neuron is the structural and functional unit of neural system. Are these points clear? Then what does neuron consist of? See, yesterday only we have completed structure organization in animals, right? So I have already discussed the structure of neuron. So what is the basic structure of neuron like? It consists of cell bodies, dendrites, and axon. The cell body contains cytoplasm and some granular bodies called Nissel's granules. Now these Nissel's granules are also present in, they are also present in the dendrites. They are also present in the dendrites. Are these clear? Are these points clear? They are also already present in the dendrites. Please let me know that these points are clear to all. Now, talking about the third part, which is the axon. Now, each branch, each end of the axon has a bulb-like structure known as synaptic knob. Okay? So here in the diagram also we see this synaptic knob, which contains synaptic vesicles, and synaptic vesicles are filled with the neurotransmitters. Is that clear? Now on the basis of number of how many axons and dendrites are present, we have three different types of neurons. See, actually there are more than these, but NCERT is just talking about three, so we'll restrict ourselves here. So multipolar. So one axon and more than two or three dendrites. Is that clear? So many dendrites, one axon, this is multipolar. So the diagram here, this represents the multipolar neuron. Is that clear? So another important thing, where are multipolar neurons found? They are found in your cerebral cortex. Bipolar means one dendrite, one axon. So they are found in the retina. Then unipolar is unipolar has only one process, and that is one axon. It doesn't have any dendrite. So they are found in the embryonic stage. So while we were embryos, our neurons were unipolar. Are these points clear to all? Are these points clear to all? Right? Okay, moving on to the next. So axons can already, so neurons, or you can say axons, can be differentiated into two further types based on whether they have this sheath or not, myelin sheath. Okay, now myelin sheath is a lipid rich, fat rich layer. If it is present, we'll call the axon as myelinated, and if it is not present, we'll call it non-myelinated. Now whether it is present or not, Schwann cells are always present. Schwann cells are going to produce myelin sheath, or they may not produce, but Schwann cells are always present. Are these points clear? But in case of brain and your spinal cord, they are not the Schwann cells. It's a different type of Schwann cell. We call it oligodendrocytes. Is that clear? So Schwann cells are present in your peripheral nervous system, not in your CNS. Are these points clear? But Schwann cells, whenever present, they may or may not produce myelin sheath. Now, gap between the two myelin sheaths is known as node of Ranvier. So this point of this place where myelin sheath is absent, node of Ranvier. Node of Ranvier, very important. Myelin sheath absent, but Schwann cell is still present. So remember, even at the place of node of Ranvier, Schwann cells are still present. See, myelinated are found in spinal cord and cranial nerves, and unmyelinated are, they are enclosed by Schwann cells but do not form myelin sheath, and they are found in autonomous and somatic neural system. Are these points clear to everyone? Please let me know if there is any doubt. Moving on to very important aspect, generation and conduction of nerve impulse. See, neurons are excitable cells. They are cells but different from rest of the cells. Why? Because they are excitable and their membranes are in a polarized state. Now, what do you understand by polarized? So there are different types of ions present across. So that is how they become polarized. Now we'll see. See, there are some channels, ion channels also on the membrane, and these channels are selectively permeable. So some channels will allow the entry or exit, entry of sodium, while others will allow the exit of potassium ions. Now the membrane at which there is no stimulus is known as the resting membrane. And remember, very important line, that resting membrane is more permeable to potassium ion. This is a PYQ. And it is impermeable to negatively charged proteins. These are these ions, sorry, these negatively charged proteins are present in the exoplasm. See, I have written it for you here. ECF, that is the fluid outside of the neuron, extracellular fluid, has more of sodium, less of potassium, while within the axon, that is the intrastitial, sorry, intracellular fluid, that will have more of potassium, less of sodium, and more of negatively charged proteins. Are these points clear to all? Now, exoplasm inside. So this is all clearly written here. Session is going to be a longer session. This is a marathon session. It will go till 11 or 11:30, and we are going to complete our entire class 11th NCERT today. Is this clear? Now, ionic gates across the resting membrane are maintained by, very important, there are some active transport of ions. We call those as sodium potassium pump. Now, very important, through sodium potassium pump, this is a PYQ. How many sodium ions go out? There are 3 sodium ions are going to go out and 2 potassium ions are going to move inside. Now this is active transport, so it means it is going to use 1 molecule of ATP. So for 1 molecule of ATP, 3 sodium goes out, 2 potassium comes in. Are these points clear? Now this is all about the resting membrane. This is all about the resting membrane. This is resting membrane. Is that clear? So resting membrane, see this diagram. The resting membrane is negative on the inner side and positive on the outer side. Are these points clear? But now, when suddenly the stimulus arrives, see, all these points are very, very important. When there is a stimulus, the membrane becomes freely permeable to for sodium ions. So that site where stimulus occurs, where stimulus arrives, that point of a membrane, that becomes freely permeable to sodium. So sodium keeps going in very quickly. So there is rapid influx of sodium, and this rapid influx of sodium is going to reverse the polarity. So earlier, which the membrane which was negative on the inside, it is going to become positive. So inner side becomes positively charged. Now this is known as depolarization. This reversal of polarity is known as depolarization, or we say the action potential has generated. Are these points clear? And this is only generation of action potential itself is known as nerve impulse. Are these points clear to everyone? Please let me know. Any break after 1 to 2 hours, we'll see. I'll give you some NEET gyan during that break. Okay? I'll give you that NEET gyan during that break. But otherwise, there is no break. We can't afford a break. How can you even ask for that? Can you explain last? Jasmine, which all lines are you asking? Please understand, unit-wise explanation will continue. That series will go on. Okay. Now, moving on to impulse. So see, so movement of this action potential from one side to another, this is impulse conduction or nerve impulse conduction. Now, stimulus-induced permeability of sodium is extremely short-lived. So the moment this rapid influx of sodium ions start occurring and the membrane becomes positive on the inner side, quickly, very quickly, there is increase in the permeability of sodium ions also. See, very clearly remember 3 things. Resting membrane is more permeable to potassium ions. Okay, note down somewhere. Resting membrane more permeable to potassium ions. Then depolarized membrane is permeable to sodium ions. And again, this repolarized will be permeable to potassium ions. Is that clear? So what happens as soon as the rapid influx occurs and the membrane becomes positively charged, the potassium starts diffusing out. Is that clear? Now, this is how the whole nerve impulse conduction is going to take place from one part to the another part of axon. Clear? Now, from one neuron to next neuron, there is transmission. So transmitted from one neuron to another is through the junctions called synapses. So there are 2 types of synapses. Remember, one is the electrical synapse and then there is the chemical synapse. So what is synapse? This is, every synapse consists of, see, presynaptic membrane or a postsynaptic membrane. But this third component, synaptic cleft, may or may not be present. Are these points clear? See here. So I'll tell you very clearly. Okay, wait a second. See, this is the chemical synapse. This is a picture of chemical synapse. So chemical synapse will have, wait. See. Okay. So this chemical synapse, wait, I hate it. When machinery doesn't listen to me, I hate it. Alright, guys. So moving on. So in chemical synapse, what happens? In chemical synapse, there are three things. One is the presynaptic membrane, then there is a postsynaptic membrane, and then there is a space in between which is known as the synaptic cleft. Is that clear? And in the synaptic cleft are released the neurotransmitters. Now neurotransmitters are going to bind onto the receptors on. Now, where are receptors for the neurotransmitters present? So, this is a very important line. The receptors for the neurotransmitters are present on the postsynaptic membrane. Is this clear? You're gonna complete class 11. Yeah, complete class 11. Are these points clear to everyone? Moving on to the next part, next page. See here. So see, neurotransmitters bind to the specific receptors. Receptors are present on the postsynaptic membrane. Now, depending upon the neurotransmitter, the potential can be excitatory or inhibitory. So there are certain neurotransmitters like acetylcholine, ACH and adrenaline which are excitatory, while others like GABA and serotonin, they are inhibitory. So depending upon the neurotransmitter, the message will be passed on. Now talking about our central nervous system. So, what is first of all, what is the function of central nervous system? Central nervous system, that is your brain and spinal cord. This is a central information processing organ. Okay, command and control system controls your voluntary movements, balance, functioning of involuntary organs, thermoregulation, hunger, thirst, circadian rhythm, everything, even your human behavior, and the function of endocrine glands as well. Also, it has sight for the processing of vision, speech, memory, hearing, intelligence, emotions, and thoughts. Now, starting with the first part of CNS, which is brain. Now, brain is protected by skull. This is this bony framework, and then it has some protective membranes known as cranial meninges. There are three types of meninges. The outer one is durameter, middle one is arachnoid, and the inner one is known as pyometer. So, which one is in touch, in contact with the brain surface? It is the pyometer. Is that clear? Then the brain has three parts. Now coming on to the brain, so outer side it is covered with skull, inner to skull there are meninges, and then inner to the meninges is the surface of the brain. Now brain in itself is divided into three parts. How many parts? Three parts: forebrain, midbrain and hindbrain. Is it clear? Let's talk about the forebrain first. Forebrain further consists of three parts. How many? Three parts: cerebrum, thalamus and hypothalamus. Now out of the three, cerebrum is the largest part of the brain, major part of the human brain. Clear? And this cerebrum is further divided into two halves known as cerebral hemispheres, but they are connected from the lower region, which is by a track of nerve fibers known as corpus callosum. See, corpus callosum is a track of white nerve fibers, and it is only present in the mammalian brain. Okay, so the layer of the cells which cover the cerebral hemisphere is the outer layer is called the cerebral cortex, and it is highly folded. Prominent folds are present. This cortex is known as the gray matter. Why? Because it is grayish in appearance. Now why is it grayish in appearance? Because it is made up of neural cell bodies, neuron cell bodies, which give the color. Now, cortex has three types of areas, very, very important: motor area, sensory area, and association areas. Is that clear? Now, association areas are going to function for intersensory associations, memory, and communication. Now, there are some tracks which are covered with the myelin sheath. Is this clear? Then, inner to that, see, these tracks which are covered with the myelin sheath, they are going to form the white matter. So there are two types of matters in your brain. Outer we have gray, inner we have white. Now the cerebrum, this larger part, cerebrum, it wraps around the small structure known as thalamus. Is it clear? What is the function of thalamus? They are the sensory and motor signaling areas. Now thalamus, below the thalamus is present a structure known as hypothalamus, written at the base of thalamus. Function of thalamus is to control the body temperature, urge of eating and drinking. There are some neurosecretory cells also in the hypothalamus which are going to produce the hormones which we have already talked about, right? The hormones for releasing hormones, then inhibitory hormones and so on. Now the inner parts of inner parts of cerebral hemispheres have structures like amygdala, hippocampus, limbic. Uh, Amygdala and hippocampus, they form your limbic system. Now, limbic system controls the emotions. So it is known as emotional brain. It regulates the sexual behavior, emotions like excitement, pleasure, rage, fear, and motivation. Clear to all? Test in the middle of class, not in the middle of the class, Neelam. I am just underlining all the lines from where questions can appear. I have already underlined every line from where the question has already come in the previous year paper. So as the thumbnail says, we are going to highlight the PYQs also. Is that clear? So questions are going to come tomorrow, because tomorrow from 19, that tomorrow is 19. So tomorrow onwards from 2 to 5, we are starting the free mock test series. Is that clear? So that mock test is going to test your performance. Now coming here to the midbrain. Where is it present? Between thalamus or hypothalamus of the forebrain and pons of the hindbrain. Now there is a special duct which passes through the midbrain, that is cerebral aqueduct. There are 4 swellings known as 4 optic lobes present in the midbrain, and these optic lobes are known as corpora, quadrigemina. Coming on to the hindbrain, pons, cerebellum, and medulla. These are the 3 parts. Pons interconnect different parts of the brain. Cerebellum is also the second largest part of the brain and also convoluted like cerebrum. And the medulla is that part of the brain which connects brain to the spinal cord. And medulla is known to participate in regulation of all the involuntary processes like respiration, cardiovascular reflexes, gastric secretions. Is that clear? Now the 3 things, midbrain, pons varolii, and medulla oblongata, they together form your brain stem. Is this clear? Okay. Now connection and form connections between the brain and the spinal cord. Please let me know, give me a thumbs up that this chapter is also quickly done. Let me know. Quick, everybody, let me know that this chapter has been well read by all of you. Let me know. Yes, please. Give me a thumbs up. Green hearts and thumbs up. Quick. Yes, please. So moving on to the next chapter of human physiology, that is chapter 17, locomotion and movement. Yes, I am running like a super fast express. Highlighted textbook, you will get the text PDF. You will get the PDF. I hope all of you have received the PDF for yesterday's session. You will get all the PDFs. Yes. Moving on to the next one. This is locomotion and movement. So see, movement and locomotion in smaller organisms like unicellular organisms like amoeba was because of streaming of protoplasm. This is a simple form of movement. While in paramecium it is because of the cilia, and then in hydra it is the tentacles which help in locomotion as well as for capturing the prey. Now very important line, all locomotions are movements, but all movements are not locomotions. How many of you are very clear with the statement? Clear? Okay. Then methods of locomotion. Now the methods will vary as per the habitat and the demand of the situation. Okay. Now for example, if we are in water, we are going to swim. If we are on land, we are going to walk, and so on. Is this clear? Now, there was this one very silly question in one of the PYQs, that locomotion, what is the motive behind locomotion? We move for the search of food, shelter, finding the mate, suitable breeding ground, and favorable climatic conditions, and to escape from the enemies. So, a little silly, but you can't miss this. Then types of movements, please see here. There are different types of movements seen in our body, like WBCs, macrophages, and other WBCs. They exhibit amoeboid type of movement, just like amoeba. Then pseudopodial movement, they have pseudopodia. Then cytoskeleton elements like microfilaments, okay, they also show a amoeboid movement. Is this clear? Then ciliary movement like paramecium is seen in our body also. So where do you see ciliary movements? Ciliary movements are seen in some of the internal organs. Can you name some? Where is ciliated epithelium present? We have learned it yesterday. Ciliated is present in your respiratory tract, in your fallopian tubes, right? This is where you see your ciliary movement. It is written, passage of ova from female reproductive tract is by ciliary movement. Then the third type of movement that is seen in a body is the muscular movement. See, all our limbs, jaws, tongue, they require muscular movement. Now, very important property of muscle is the contractile property, which allows the movement. Is that clear? Now, locomotion, very important. This is a PYQ line. Locomotion requires a perfect coordination of three systems. Now, what are the three systems which work in tandem for making the locomotion or movement possible? One are your muscles. Muscles are attached to your bones, so skeletal system, and they both move because your brain is signaling them. So muscular system, skeletal system, and neural system. These are the three systems which work in tandem for movement. Are these points clear to everyone? Are these points clear to everyone? Okay, moving on to muscle now. Let's talk about muscle. By the way, there is fourth type of movement also seen in a body, which is flagellar movement. Can anyone name me in the chat? Tell me in the chat which cell in the body shows a flagellar movement. Yes, please. Which cell in the body shows the flagellar movement? Quickly. I'll give you a hint. It is the smallest cell in the body. Moving on to the muscle now. So cilia and flagella are the outgrowths of the cell membrane. See, flagellar movement very clearly written in front of you is spermatozoa. So sperm shows that flagellar movement. Also, we have seen that the sponges have collar cells and they are flagellated cells. So water movement in the canal system of sponges is because of flagellar movement. Then euglena has two flagella, but the one is useless. Only one helps in the locomotion. So muscle is mesodermal in origin. 40 to 50% of the body weight is because of muscles. The very important special properties of muscles are, you have to remember it as 3E plus C. So 3E means excitability, extensibility, and elasticity, and C stands for contractibility. So it can be excited, it can be extended, it can be contracted, and this property it can regain the shape is known as elasticity. On the basis of the locomotion, guys, there are three types, based on location, there are three types of muscles. We have already discussed them yesterday in detail. That what are the features of skeletal, involuntary, and cardiac muscles. So let's see. Skeletal muscles, also known as striated muscles, voluntary in nature, help in locomotory movements and changing of body postures. While visceral muscles, also known as smooth muscles, they are present in the viscera. That means the internal organs, inner walls of hollow visceral organs, alimentary canal, reproductive tract. Also known as smooth muscles or non-striated because they don't have any striations. They are not under your will, so they are involuntary, and they help in the transportation of food through digestive tract and gametes through the genital tract. Are these points clear to all? Please let me know. Yes, yes, yes. Now, as the name suggests, cardiac muscles, where are they supposed to be found? They're supposed to be found in the heart. Okay. Now, they have branching pattern. See, skeletal and non-skeletal, striated and non-striated, they are not branched. But cardiac muscles are branched. Is it clear? They show faint striations also, but they are involuntary in nature. So, isn't it that cardiac muscles are functionally more like involuntary, but structurally more like voluntary? Is that clear? So the nervous system does not control their activities directly. So we know that our heart is myogenic, autorhythmic, but so the rhythm has to start by the heart itself, but nervous system can influence it. So that is why it is written the nervous system does not control their activity directly. Is this point clear? Then moving on to the structure of the muscle. See, skeletal muscle is made up of, one muscle has many muscle bundles. See the diagram also. Muscle has many muscle bundles. Do you see this? Now bundles are also known as fascicles. Now the covering around the bundle is known as fascia. So it is formed of collagenous connective tissue called fascia. Is that clear? Now muscle bundles have these small muscle fibers. Every muscle bundle has so many muscle fibers, and this fiber is equivalent to the muscle cell. Are these points clear? Now if I talk about the muscle cell or the muscle fiber, then we are going to use the same terminology which is used for a cell. If it is a cell, it will have a plasma membrane. So the plasma membrane of muscle fiber is known as sarcolemma. Cytoplasm is known as sarcoplasm. Okay. Now fiber, as we have read yesterday, that skeletal muscles are multinucleated. This multinucleated condition is known as syncytium. Is this clear? The endoplasmic reticulum is known as sarcoplasmic reticulum. So you're going to use the prefix sarco for muscle fiber. Is that clear? And this endoplasmic reticulum, this is of smooth type. This is SER, and it is a storehouse of calcium ions. See, I'm already revising so many things with you. Make sure that you are writing down these points. Is this point clear? All right, moving ahead. Now, The characteristic feature of muscle fiber is a large number of parallelly arranged myofilaments or myofibrils, which has alternate light and dark bands. Now, there are two types of proteins present in the muscle. One is actin and second is myosin. These are the contractile proteins. Now, light band contains actin and is called I band or isotropic band, while dark band or anisotropic band contains both. It majorly contains myosin, but actually it has both actin and myosin. See, both proteins are arranged as rod-like structures parallel to each other. Now, actin filaments are thinner compared to the myosin filaments and known as thin and thick filaments. So, thick because it is thick, myosin is thick, actin is thin. Now, they form a particular arrangement which is known as a sarcomere. Now, see, the sarcomere is present between the two Z lines. So, I band is an elastic band called Z line. Sorry, each I band is an elastic fiber called Z line which bisects it. In the center of I band is an elastic fiber. So Z line is made up of which type of fiber? Elastic fiber. Where is it present? In between the I band. What is the function? It bisects it. Now this line is a PYQ. Please remember. Now thin myofilaments are firmly attached to the Z line. So see this diagram here directly. Just see this diagram. So this is the structure of a sarcomere. Do you see? Sarcomere extends between two Z lines. These dark black colored lines are the Z lines. So these peripheral proteins are actin. Okay, these are the thin filaments basically, majorly made up of actin. And this dark red filament. This is the thick filament made up of myosin. Okay. Now, as far as this myosin can extend, we have A band, and then the peripheral area represents the I band. Now, can you see the I band is being dissected by the Z line? Can we see it? Why don't I teach in bilingual? Because this is a NEET English channel. Isn't that so? This is particularly for those students who are always complaining and suggesting me, ma'am, please teach in English. So here I am this time. I've taught in bilingual language for enough time. All right, coming on to now H zone. So see, within the A band, the area which is not getting overlapped by thin filaments, this central area is known as ZH zone. This is known as H zone. Now talking about the contractile proteins, which are the two contractile proteins? Actin and myosin. So each actin filament or thin filament is made up of two F filaments, filamentous actins. Thus, how many? See, thin filament is going to have two filamentous actins, and filamentous actin is further made up of G actins. Is this clear? Other than this actin, F actin, we have two bands of tropomyosin, and at regular intervals we have a complex protein known as troponin. Now troponin is further made up of three elements: TPC, TPI and TPM. Is this clear? Whereas myosin is a polymerized protein and made up of the monomers known as myosin. See here. So everything will be clear from the diagram. Wait. Let's come on to this diagram. So guys, this diagram of sarcomere is very, very important. By the way, sarcomere is known as the unit of contraction. I hope you know that. Is that clear? Okay. Now moving on to the next part. See, we have already read this. So every meromyosin has two parts. One is the globular head with short arm, and arm and head together form the heavy meromyosin, and there is this long tail which forms the light meromyosin. Is that clear? So see this diagram. So we have troponin, tropomyosin, F actin. These three proteins are present in the thin filament, while this snake-like structure, this is the meromyosin. This is a monomeric unit of the thicker filament, which is the myosin. Now each meromyosin has this long tail, which is the light meromyosin, and the head and the cross arm, and together head and the cross arm forms your heavy meromyosin. Now head has these two sides, very important. This is a PYQ. Head has two sides, actin binding site and ATP binding site. Is this clear? Then moving on to the mechanism of muscle contraction. Now see, all the mechanisms should be well learned in a proper sequence, because you can get questions based on arrange the following steps in the correct sequence. Is this clear? Okay. Shri, we are going to discuss the third unit, which is cell unit, cell division and biomolecules. Okay, moving here. So mechanism of muscle contraction, it can be explained by this theory of sliding filament theory. Now what is the underlining statement of this theory? That thin filaments are going to slide over the thick filaments. Remember, it is the thin filament which is going to slide over the thick filament. Just in order to remember, see, it is difficult for a thicker person to move very easily, right? So the thick is going to be static and the thin is going to move. Is this point clear? Okay, moving on to the mechanism exactly. So it is initiated by a neural signal by the central nervous system. And how does central nervous system send a signal to the muscle? Through a motor neuron. Now the junction between the neuron and the sarcolemma is known as neuromuscular junction. Is this clear? Or the motor end plate. This is where the neurotransmitters are going to be released. Now, as soon as the neurotransmitters are released or action potential is generated, it is going to be passed, it will be passed on to the SER, sarcoplasmic reticulum. Remember, we said it is a storehouse of calcium ions. Now that SER is going to release the calcium ions, and they are going to bind with one subunit of troponin. Is that clear? Now, as soon as calcium binds with this troponin, it is going to unmask the site of myosin. It is going to unmask the site of myosin. Is that clear? Now, energy from ATP, now ATP will break down, ATP will hydrolyze, and that is going to release energy for muscle contraction. So, as soon as ATP is hydrolyzed, this head of myosin will now bind to the actin. Is this clear? See here in the first step, ATP's hydrolysis is taking place. Then this energy will be used for the binding of myosin to the actin. This actomyosin formation is known as cross bridge. What is cross bridge? Binding of actin and myosin. Clear? Then there is sliding rotation. Now this myosin head moves backward and carries the thin filament along. So that is how thin filament is going to slide over the thick filament. So later on, ATP will be regenerated and the head of the myosin will detach from actin, and this is known as breaking of cross bridge. So this whole represents the stages of cross bridge formation or the mechanism of muscle contraction. Are these points clear to all? Now very important that during the muscle contraction, what happens to the sarcomere? So you have to remember this. So shortening of sarcomere takes place. Basically Z lines come closer, shortening of the sarcomeres take place. Now in this, I band is going to be reduced, but A band remains the same. Is this clear? So what all happens? Z lines come closer, your sarcomere shortens, your I band shortens, but A band remains the same. Are these points clear? Please let me know that these points are super clear to all of you. Yes, please. Please let me know that these points are super clear to all of you. Is there any way to improve botany? See, the way to improve your performance in every subject is now to solve questions. Okay, so that is why mock series is coming up this from this Sunday, tomorrow onwards, 2 to 5 p.m. Is this clear? And then keep reading, keep underlining these important words, and then unit wise revision is also going on. Is that clear? All right, moving ahead, coming on to the next part is, see this diagram. Do you see? This is the original, this is the relaxed state of sarcomere. Now when it has contracted, do you see that Z lines are coming closer? The sarcomere has contracted, sarcomere has shortened. So shortening of sarcomere is going to lead to muscle contraction. See here. Now, once the muscle has contracted, it must come back to the state of relaxation. So for that, calcium ions go back to the sarcoplasmic cisternae. Cisternae, as we know, is the component of SER, right? And when calcium will be removed from troponin, it will go back into the SER. Then actin filaments will be masked. So the binding place for myosin will be masked again. Z lines will go back to the original position, and this is known as relaxation. Now, repeated activation of muscles, so repeated contractions will lead to accumulation of lactic acid, and this is due to anaerobic breakdown of glycogen, and this leads to fatigue, or we call it cramps. Okay, so muscle fatigue, which leads to muscle cramps, it is because of anaerobic breakdown and due to accumulation of lactic acid. See, every muscle has a red colored oxygen storing pigment known as myoglobin. Every muscle has. But there are certain muscles which have more of myoglobin, so they appear red in color. Which have less of myoglobin, they will appear white in color. So see, high in myoglobin, we call them red fibers, and they have plenty of mitochondria also. So they use more of oxygen also. So they can respire aerobically for a longer period of time, and that is why we call them aerobic muscles. Red fibers, more myoglobin, more mitochondria, more aerobic respiration. But they have less of calcium and less of sarcoplasmic reticulum. While on the other side, the fibers which have less of myoglobin are whitish in color, so they will have less of mitochondria, less aerobic respiration, so anaerobic process of energy will be produced. But they have more of SER and more of calcium. Are these points clear to everyone? Please let me know. Please let me know. What should I repeat? Please go with the flow. If you have missed on any chapter, yes, if you have missed on any chapter, you can always watch the, of course, the videos. You can watch the recorded video later on. But now let's just go with the flow. Now moving on to the skeletal system. So our skeletal system is majorly made up of bone and cartilage, and both of them, they constitute the specialized connective tissue. Now while bone is very hard because of presence of calcium salts, your cartilage is slightly pliable due to chondroitin salts. Now how many bones do you have? 206. These 206 bones can be divided into axial skeleton with 80 bones and rest of the 126 bones in the appendicular skeleton. Now what are the components of axial skeleton? Your skull, your vertebral column, your sternum, your ribs, they constitute your axial skeleton. Now, this diagram is going to show your skull bones. Now, all those which have been highlighted in blue. See, ethmoid bone, 1 in number, sphenoid bone, 1, frontal, 1, parietal, 2, temporal, 2, and occipital. These are 8 bones which form your cranium. They form your cranium, that means the brain box. While rest of the bones written here, these are the facial bones, but not all have been represented here. So see, 8 bones in the cranium and 14 bones in the facial region, the face, together form 22 bones. So both face and your cranium constitutes your skull. Are these points clear? So how many bones in your skull? 22 bones. Remember, now hyoid is not a part of skull. Okay, it is a U-shaped bone present at the base of the buccal cavity. So you can't feel your hyoid bone from outside actually. It is a horseshoe shaped bone. Now facial bones I've told you 14. Now each middle ear will have 3 bones, malleus incustate. So in total we have 3 pairs or 6. These bones are collectively known as ear ossicles. So skull articulates with the upper part of your vertebral column, that is your backbone, with the help of 2 condyles. And we have 2 condyles, so that is why our skull is known as dicondylic skull. Is this clear? Amphibians and mammals have dicondylic skull. Clear? Then vertebral column or your backbone is further made up of 26 small bones. Now where is your vertebral column present? On the dorsal side of the body. It is dorsally placed. Now the center of each vertebra is hollow, and that is the place for the movement or for the passage of spinal cord. So that central space in each vertebra is known as neural canal. So see, this is canal. This, let's say my hand represents a vertebra. So the canal is known as the neural canal, and through this neural canal, your spinal cord is going to pass through. Now, these 26 bones have been differentiated into 5 regions. 7 in the neck region called cervical, 12 in the thoracic, 5 in the lumbar, 5 fused to form 1, that is the sacral, and 4 fused to form 1 coccygeal, that is the tailbone. Now, it is an important line written in your evolution chapter that all mammals have 7 cervical vertebra. Is that clear? Okay, it's written here also. Yes. The number of cervical vertebra is 7 in almost all the mammals. Now, vertebral column, what is the function of vertebral column? Apart from giving the shape to your body, and it is majorly to protect your spinal cord. Are these points clear? Are these points clear? Yes. So, sternum. Now, where is sternum present? It is present on the ventral side. This flat bone you feel in the center of the chest is the sternum. Flat bone in the ventral midline of the thorax. Are these clear? Then, how many ribs are present? 12 pairs of ribs. Overall, 24. Now, these 12 pairs are differentiated into three parts: true ribs, then false ribs, and floating ribs. See, ribs are bicephalic. Why are they bicephalic? Because there are two surfaces to join. One surface joins with the backbone or the vertebral column, and the other is going to join with the sternum or or to the other rib. So since there are two articulation surfaces, we call them bicephalic. Now, true ribs are first 7 pairs because they are directly attached to the sternum, dorsally to the vertebral column, and ventrally to the sternum. First 7 pairs. Do you see here in the diagram also? Then we have 8th, ninth, and 10th. These are known as false ribs, dorsally attached to the vertebral column. In front, they are, or in the ventral region, they are attached to the 7th vertebra. Sorry, not vertebra, 7th rib. They in the ventral region, they are attached to the 7th rib. Is that clear? We also call them vertebrochondral. Now, they are not directly attached to the 7th rib, but through the hyaline cartilage. So that is why we call them vertebrochondral. Vertebrochondra word is for cartilage, so vertebrochondral. And then last two, 11th and 12th, not connected ventrally. Not connected ventrally. So they are just floating, and we only call them vertebral ribs. Is that clear? So now vertebrae, that is the backbone, your ribs, and the front bone, that is the sternum. Together these three form your rib cage. So now can you calculate how many bones are present in the rib cage? Just calculate. 26 vertebrae, then 24 ribs, total 50, and one sternum. So there are 51 bones in the rib cage. Are these points clear? Now coming on to the appendicular skeleton. Appendicular skeleton consists of your limb bones and your girdles. Is that clear? Each limb is going to have how many bones? 30. 30 in the arms, 30 each arm, 30, and each leg has 30 bones. Is this clear? Now see the diagram here. See the diagram here. This represents your forelimb. So the bones present in the forelimb, your humerus, radius, ulna, all one each. Then your 5 carpals. Sorry, you have how many carpals? We have for yes, please. Okay. Yeah, so we have 1 radius, 1 ulna, we have 8 carpals, yes, and then we have 5 metacarpals, and we have 14 phalanges, right? So we have radius towards the thumb side and ulna is towards the little finger side. The wrist will have carpals. How many? 8. So carpals are the wrist bones, metacarpals are present in the palm area, and your fingers have the phalanges, 14 phalanges. Is this clear? So in total they form 30 bones. Now very important that this head of humerus, where does it fit in? It fits in with the scapula, in the cavity of the scapula. That cavity is known as glenoid cavity. Are these points clear? Then talking about your lower limb also has 30 bones. Clear? Now, what are those bones? Femur. Femur is your thigh bone. Then we have patella in between. See, elbow doesn't have a separate bone, but knee joint has a separate bone, which is patella. Then tibia, then fibula, then tarsals. Now, wrist has 8 bones, but tarsals have 7 bones. Is it clear? Then metatarsals will be 5 again, and phalanges will be 14 in number. Is this clear? Ma'am, can we call the last 2 pairs of bones bicephalic? They're not joining with anything in front, but otherwise there are 2 ends, right? Okay. Now coming on to here. So patella, it says it is a cup-shaped bone which forms the kneecap, and it is present on which side? It is present on the ventral side. Clear? Then pectoral girdles. So limbs we have talked about. So each limb has 30 bones, in total 120. Now what about the rest of the 6 bones? So we have shoulder girdle, which is called the pectoral girdle. It is made up of two pairs. One is clavicle and scapula, and then pelvic girdle, which forms the hip region. It has two bones called the coxal bones. So see here. So pectoral girdle will have scapula and a clavicle. Scapula is a triangular flat bone dorsal part between the second and the seventh rib. You have to remember this. And the slightly elevated ridge of scapula is known as spine, and which projects as a flat expanded process called acromion. And it is through acromion, below the acromion is present a cavity where humerus is going to fit in, and we call it the glenoid cavity. Now, what is the purpose of acromion process? Acromion is the place where your collarbone is going to attach. Your collarbone is going to attach with the acromion process. Is this clear? And on the ventral side, it is going to attach to your sternum. Is this clear? Now, then moving on to the pelvic girdle, the hip girdle, it has two coxal bones. Now each bone will be formed of, see here, this coxal bone, this bone. Each coxal bone is formed of three regions: ilium, ischium, and pubis. Now the point where all these three regions attach, the point of fusion of these three regions, ilium, ischium, and pubis, is known as acetabulum, and this is where your femur fits in. Your femur bone, the head of the femur, fits into the acetabulum. Is it clear? And the region where the two coxal bones fuse, that is known as pubic symphysis, and this is where fibrous cartilage is present. Now moving on to the very important part of this chapter is joints. So we have three types of joints: fibrous joints where no movement occurs. For example, in your flat skull bones, they form sutures. The joints between your skull bones are known as sutures. And what type of tissue is present here? That is dense fibrous connective tissue. There is no movement here. Then second type is cartilaginous joints between adjacent vertebrae, and this is where limited movement is allowed. So between the two vertebrae, cartilaginous joints are present. Is this clear? Then the third type, and this third type of a joint, that is synovial joint, has synovial fluid in between, and this fluid allows a considerable amount of movement. So fibrous, no movement; cartilaginous, limited movement; and synovial joint have free movement. Now synovial joints are further of different types. Now these examples are super important. Ball and socket joint, the example is humerus and pectoral girdle. So your humerus and pectoral girdle, where this movement is allowed, this is called ball and socket. And second is between femur and acetabulum. Then hinge joint is your knee joint and as well as your elbow joint. Pivot joint is between the first two vertebra. The first vertebra is known as atlas, the second is axis, and they together constitute the pivot joint. Gliding joint, where the two bones are going to slip on each other. They are present in the carpal and in between the tarsals. And then we have the saddle joint. This is present between the carpal and metacarpal of the thumb, which allows this movement. Is this point clear to all? Right? Very good. So I have given you some extra examples also. See, I am reading NCERT, but when I discuss unit-wise revision, when I do unit-wise revision, I will be giving you some extra examples as well. Clear? Please let me know that these points are clear. Now these disorders are very important. All these disorders have come in PYQ some or the other time. Now first of all, myasthenia gravis. Remember, myasthenia gravis is autoimmune disorder at where antibodies are going to attack on the neuromuscular junction, leading to fatigue, weakening, and paralysis. While muscular dystrophy is a genetic disorder and leads to progressive degeneration of skeletal muscles. Tetani is rapid spasm, wild contractions, which are because of low calcium ions. And then we have arthritis, inflammation of joints. Osteoporosis, very important, is age related, where the bone mass decreases, which increases the chances of getting fractures, and it occurs due to decreased level of estrogen. Why decreased level of estrogen? Because low estrogen will decrease your calcium absorption. Is it clear? So as estrogen decreases, calcium absorption increases, the bone mass decreases, and this happens because of age, especially in females because of menopause. Gout is again inflammation of joints due to accumulation of uric acid crystals. Is that clear? So gout is a metabolic disorder due to uric acid accumulation. Please let me know that these points are clear to all. Are these points clear to all? Please let me know. So guys, with this we have completed third chapter, that is muscular that are movement and locomotion. And moving on to the fourth chapter of human physiology, which is excretory products and their elimination. Quickly. So, first thing, ammonia is the most toxic form, uric acid is the least toxic form. Remember that. Now, which all organisms are ammonotelic? Many bony fishes. Remember, these examples are very, very important. See, still there is time. You can write down these examples in the white space. Always write down these important things separately. Okay, a monotelic are the which are the examples? Many bony fishes, aquatic amphibians, and aquatic insects. They are monotelic. Why? Because they have lot of water, and ammonia requires large amount of water. See, for its elimination. Is it clear? It dissolves readily in water and excreted by diffusion through body surface or through gill surface. Then let's talk about uric acid, the least toxic. It requires least amount of water, and then we have intermediate, which is urea. So urea is released by mammals. We release urea, right? Mammals, many terrestrial amphibians, and marine fishes. So many marine fishes excrete urea, while when it is clearly written bony fishes, so many bony fishes we know are freshwater and marine. So many bony fishes are going to secrete ammonia, while marine fishes are going to secrete urea. Are these points clear to all? Now, uric acid is secreted by those organisms which have very less water. So, in order to conserve water, reptiles, birds, land snails, insects, they release uric acid in the form of pellet or paste. Because uric acid, being least toxic, can be stored in the body. Are these points clear to all? Yes, we are going to do the whole book today. Whole book. Coming on to invertebrates. So what are the excretory structures? Not all have kidneys, right? So invertebrates have simple tubular form. See, we have already discussed animal kingdom. So you already know that different phyla have different types of excretory structures. Okay, so this is just a glimpse of it. Vertebrates have complex tubular organs called kidneys. We have also discussed that there are Malpighian tubules present in arthropods, in insects. Then antennary glands or green glands are present in crustaceans, which are again arthropods. Then coxal glands are also present in arthropods. I hope you are able to recall. Then flatworms have flame cells or protonephridia. Do you remember? Yes or no? Protonephridia? Nephridia in case of earthworms or annelida? Yes. So different organisms will have different structures. So can we get the PDF of highlighted PDF? Yes, you can, but it's better if you underline it along with me. See, work which is self done is always better, right? Okay, moving on to human excretory system. So first of all, talking about the kidney, we have a pair of them. And where are they present? Very important. Between the last thoracic vertebra and third lumbar vertebra, close to the dorsal inner wall of abdominal cavity. Just remember last thoracic and third lumbar vertebra. Clear? Then there is this place called this hilum, through which ureter comes out. Then the renal pelvis, the broad region, where projections called calyces open. Then the covering of kidney is known as capsule. Then every structure can be divided into outer and inner region, right? And the outer region will be known as cortex and inner region will be known as medulla. Are these points clear? Then medulla will be divided into medullary pyramids. Medulla will be divided into medullary pyramids. Are these points clear? Please let me know. So let's go to this diagram directly. See, this is the capsule, the covering of kidney. Outer region is known as cortex, inner region is known as medulla. Now medulla has these triangular structures known as medullary pyramids. Clear? The ducts are known as calyces. All these ducts open in a broad region known as renal pelvis. Clear? Now, from pelvis arises the ureter. So the region, the notch from where ureter comes out or the blood vessels enter or exit, that region is known as hilum. Is this clear? Now, very important. There are regions where there are portions where cortex is going into the medullary region. Do you see that? Do you see that? The cortex enters in between the medullary pyramids. Now, these regions are known as renal columns or columns of Bertini or columns of Bertini. Is this point clear? Are these points clear to all? Clear? Now, if, now the unit of kidney is known as nephron and every kidney has 1 million nephrons. These are the functional units. Every nephron has two parts, glomerulus and the renal tubule. Now, renal tubule is further differentiated into Bowman's capsule, PCT, DCT, and loop of Henle. Are these points clear? PCT, DCT are highly convoluted, right? Can we arrange it in the sequence? So we have glomerulus, that is one part. Second is the renal tubule. Now, renal tubule, the first part is Bowman's capsule. Then we have PCT. Then we have U-shaped loop of Henle, and then we have DCT again. And many DCTs open in collecting tubule. Are these points clear? So the loop of Henle has two limbs. This is the descending. The one which will carry the filtrate downwards is the descending limb, and the one which will carry filtrate on the upward direction is known as the ascending limb. Are these points clear to everyone? Please let me know. Please let me know. So where are exactly these things arranged within the kidney? So by the way, Malpighian corpuscle. What is Malpighian corpuscle? The term Malpighian corpuscle is used for Bowman's capsule along with the glomerulus. So what you see in here is the Malpighian corpuscle. Is that clear? So Bowman's capsule with glomerulus is known as Malpighian corpuscle or even renal corpuscle. So this PCT and DCT, they are in the cortical region, while loop of Henle goes deeper into the medulla. Now there are two types of nephrons. The nephrons which don't have a long loop of Henle will be majorly present in the cortex area. We'll call them cortical nephrons. Is that clear? So they have a very small loop of Henle. It is very short. While the nephrons which have a long loop of Henle runs deep into the medulla, so it covers both cortical and medullary region. They are known as juxtamedullary nephrons. Are these points clear? Now, actually, we have more of cortical nephrons, around how many? Let's say 80% of the nephrons are cortical nephrons, and only 15 to 20% are juxtamedullary nephrons. Are these points clear? Now, when a nephron has a long loop of Henle, then it also has another structure which is called vasa recta. So the U-shaped loop of Henle has a U-shaped blood vessel, U-shaped capillary network also, which is known as vasa recta. Now see, it is clearly written vasa recta is absent or highly reduced in the cortical nephrons. Are these points clear? Are these points clear? Please let me know. Okay, then moving on to urine formation. See, very, very important. So first step of, see, urine formation occurs in three steps. One is ultrafiltration or glomerulofiltration. Second is reabsorption. Third is secretion. Clear? So first step is glomerular filtration, where filtration occurs in the glomerulus. So how much blood is filtered every minute? Remember, 1100 to 1200 ml of blood is filtered every minute by the kidneys, and this is around 1/5 of the total blood. What is the total amount of blood that you have? That is 5,000 to 6,000. So isn't 1100 to 1200 constituting 1/5 of the total? So every minute 1/5 of the blood is going to be filtered through your kidneys. Is this clear? Then filtration of blood requires three membranes. So this filtration membrane is made up of membranes, three layers. One is the endothelium of glomerular vessels, then epithelium of the Bowman's capsule, and we know that epithelium tissue is always going to have a basement membrane. So there is a third basement membrane. Please let me know that these points are clear to all of you. Please tell me. Now, the epithelial cells of the Bowman's capsule have podocytes. These are the special cells you will see only in the Bowman's capsule. Clear? And then there are some spaces between the podocytes. These spaces are known as filtration slits or slit pores, and it is through these slit pores that filtration takes place. Please let me know that these things are clear to all. Almost all the constitute. So what all things get filtrated? So almost everything from the blood comes out. See, why do we call it ultrafiltration? We call it glomerulofiltration because it occurs in the glomerulus. Why do we call it ultrafiltration? Because it occurs under very high pressure. It occurs under very high pressure. And now this high pressure is going to throw out everything from the blood. Is it clear? Except for two things. The cells of the blood will not come out through the glomerulus, and second, the blood proteins. See, it's written almost all constituents of plasma except the proteins are going to pass out into the lumen of the Bowman's capsule, and this process is known as ultrafiltration. Why? Because occurring under very high pressure. Please let me know that these points are clear to all. Please let me know these points are clear to all. It Intira Kirthana, what? Okay, I'm not known to this language. What language is this, and what are you trying to say? Can we all communicate in English, please? I just know three languages: Hindi, Punjabi, and English. Class will continue till we complete our chapter. Sorry, not a chapter, till we complete our book of class 11. So as you have seen it on the thumbnail, this is class 11 complete biology division, and we have underlined all the PYQs. So if you go through this highlighted PDF, you will not have to go through the PYQs. They're all in this book. He is speaking Telugu. Okay. Okay, Telugu. Cool. I don't know Telugu. I don't know any South Indian language. Anyways, guys, let's all talk in a common language that even I can understand. Okay? Cool. Now, moving on to the amount of filtrate. See, how much filtrate does the kidney produce per minute? This is known as glomerular filtration rate. And this is 125 ml per minute. So, every minute, just remember, underline, see, let's revise once again if there is any confusion. 1100 to 1200 ml of blood passes through the kidney every minute, but out of that 125 ml per minute is the amount of filtrate produced. So in total 180 liters per day. Is it clear? So filtrate produced per minute is known as GFR, glomerular filtration rate. Is that clear? I've also given you one mnemonic as GFR, which is for the regions of adrenal cortex, if you remember. Okay? Yeah. Then moving on to JGA. See, JGA, juxtaglomerular apparatus, sensitive region in the DCT tubule and formed by the modifications in the DCT and the afferent arteriole at the location of their contact. So where DCT and afferent arteriole are in proximity, that is where this JGA is produced. Juxtaglomerular cells are present. Is that clear? Sorry, juxtaglomerular apparatus is present. Clear? Now, this JGA is known to produce renin, which stimulates the blood flow, and ultimately, this is going to help in decreasing the, increasing the blood pressure. So we have seen that in one day, 180 liter of filtrate is produced, but only 1.5 liters of urine is produced. So that means 99% of the filtrate will have to be reabsorbed by the process known as reabsorption. So that is the second step of urine formation. Now, reabsorption occurs by active mechanism as well as passive mechanism. So the things which are reabsorbed actively, remember, glucose, amino acids, and sodium ions, they will be reabsorbed actively. While the nitrogenous waste, that is urea, will be reabsorbed, or uric acid, it will be reabsorbed by passive transport. Even water will be reabsorbed by passive transport. Is this point clear? And then we have third step of urine formation, which is secretion. So secretion means the things which have not been taken out of the blood will be actively pumped out of the blood. For example, H positive, potassium, and ammonia into the filtrate. So understand that in organisms where glomerulus is totally absent, tubular secretion is the only step in urine formation. Is this clear? Are these points clear? Yes. Urea, uric acid, and water, all passively. Okay. Moving on to now functions of the tubule. So PCT, which is the renal tubule, first part of the renal tubule. PCT is the place where maximum reabsorption has to take place. So to increase the surface area, it is lined by simple cuboidal brush bordered epithelium. We have just read the chapter of animal tissues yesterday, right? 70 to 80% of the electrolytes and water are reabsorbed. So where is maximum water reabsorption going to take place? PCT. Maximum sodium reabsorption? PCT. Everything PCT. Is it clear? Maximum reabsorption. While there are certain things which are 100% reabsorbed in PCT itself, for example, amino acids, for example, glucose. Then what about loop of Henle? Reabsorption is minimum here. in the ascending limb particularly. Okay, the descending one is permeable to water, ascending is permeable to the electrolytes. These lines are PYQs, very, very important. These lines are PYQs. Is that clear? Guys, I feel those of you who are just concentrating now, who are just focused, they should not read chat. You should come to the chat only when I ask you to just give me a thumbs up or ask your doubt. Otherwise, you should be busy highlighting what is written, what I have highlighted, or revise or read with me. I know it is difficult to read NCERT. Can you read NCERT in 5 hours yourself? So this is the way you can read NCERT. And even better, when the lecture is done, you can revise it at higher speed. Just increase the speed to 1.5 or 2x and you can revise it even better and even faster. But revise once with me. Now, loop of Henle, okay, the descending one is permeable to water and the ascending one is permeable to electrolytes. Is it clear? While DCT shows conditional reabsorption. So normally DCT will not show reabsorption only if it is needed. By chance there is some shortage of sodium ions or water, then that reabsorption will continue. So it is conditional, it is facultative. So it is capable of reabsorption of bicarbonate ions and selective secretion of water and potassium. Are these points clear? Now this diagram is super duper important. So if you remember the exact picture of this diagram, then you can easily understand that from which region reabsorption and secretions are taking place. So the arrowhead which is going away from the nephron, it represents what? Reabsorption. So all the chemicals written on the top of the picture, they represent being reabsorbed, while the arrowhead towards the nephron represents secretion. So from PCT, can you see what is being reabsorbed here? Bicarbonate, NaCl, water, nutrients, potassium, and the secreted things are H positive and ammonia. Are these points clear? Okay. Similarly, see, descending limb is permeable to water, ascending is permeable to NaCl, while in ascending, urea is also being reabsorbed. Now, collecting duct, where is collecting duct present? Collecting duct extends from cortex to inner part of the medulla and reabsorption will continue, again only facultative. Small parts of urea from interstitium will be present in the interstitium to keep up the osmolarity. So very, very important question, and it is a PYQ. Listen, which are the two things which maintain osmolarity of the interstitium? One is urea, second is your sodium. Is it clear? These are two, but particularly urea. Is that point clear to everyone? Please let me know. Now coming on to mechanism of concentration. So filtrate can be concentrated further by counter current mechanism. Okay, for that counter current mechanism, we need two structures. One is loop of Henle and second is vasa recta. And remember, this ability is only present in mammals to produce a concentrated urine. Is this clear? Then see, we are starting from backwards, so there are still many chapters left. But I think this human physiology is going to take the maximum time. Now, see, increasing osmolarity towards inner medullary interstitium. So, osmolarity goes on increasing as you go deeper into the interstitial area. So, it starts from 300 and goes up to maximum 1200 milliosmoliters. So, already told you what are the two things which maintain that gradient and the osmolarity. It is sodium chloride and urea. Is this point clear? Now, this diagram is important and we will explain it more during our unit-wise revision. Clear? Now, coming on to the regulation of kidney functions. See, hypothalamus, JGA and heart. These are the three structures which will be busy in regulating the kidney function. So, as we know, hypothalamus is going to produce ADH, which will facilitate reabsorption of water from later parts. What are the later parts? DCT and CT, right? And prevents diuresis. So whenever there is increase in the body fluid, an increase in the body fluid volume, it suppresses ADH. See, increase in the body fluid means high blood pressure. Okay, it will result in high GFR and high blood pressure. Then, how does it do that? So, JGA, that is juxtaglomerular apparatus, has juxtaglomerular cells which release renin. Renin is going to convert, so renin comes from your kidney. Which cells? JG cells. Now, in your blood, renin will be released in your blood. Now, in your blood is present angiotensinogen. This angiotensinogen was produced by your liver and it converts angiotensin 1 into angiotensin 2. Is this clear? Renin converts angiotensinogen into angiotensin 1, and then angiotensin 1 is later converted to angiotensin 2 in the presence of ACE. Are these clear? Now, angiotensin 2 is a powerful vasoconstrictor, and when it is going to constrict the blood vessel, the blood pressure will automatically increase. Yes. Also, angiotensin 2 is going to stimulate your adrenal cortex to produce aldosterone to reabsorb more sodium ions. Is that clear? So that is why we call it RAS mechanism. Renin, angiotensin, aldosterone system. Why? Because there are three heroes in here. It is a multi-star movie. See, renin, angiotensin 2 and aldosterone. These three together constitute a RAS mechanism. Are these points clear? Okay. Then what if the blood pressure is already high? How is kidney going to regulate that? So for that, heart is going to come in. So the walls of atria, the atrial walls of heart are going to produce ANF, which will dilate your blood vessels. So when the blood vessels get dilated, area increases, pressure decreases. So basically, ANF and RAS mechanism, they are antagonistic to each other. That is why it is written, NCERT is written, ANF mechanism checks on the renin-angiotensin system. Are these points clear? कुछ समझ नहीं आ रहा? How is that even possible? We are reading NCERT. If you have already prepared enough for your NEET, guys, this is a quick revision. This is not for the newcomers. This is for those who are going to appear in NEET 2026, right? And in case you have missed on any part, wait for a second. You can go with me, go with the flow. As soon as I start the next chapter, nothing to panic. I feel, you are in such a sensitive zone right now, all of you, and I totally understand that. I totally, even today, I will be in the similar state if I have to take any exam. We are always so sensitive near the exam that even the slightest change can just make us go anxious, right? Don't go anxious right now. Relax. Take a deep breath. I have started since 7:30. So those of you who were attending the other lecture, no worries. You can attend the chapters which you have missed in the recorded video. Okay? Okay. Now moving on. Let's see. Micturition. See, micturition is passing out of urine. So what is the mechanism of micturition? Now that was a PYQ. What is the correct sequence of micturition? See, your urine is stored in your urinary bladder, right? Now a voluntary signal will be sent by the central nervous system. So signal will be initiated when your bladder is stretched, right? There are stretch receptors present on the walls and they send signal to CNS, and then CNS will again send back the signal for the contraction of muscles of the bladder. So bladder is going to contract, but the urethral sphincter is going to relax. Now this will lead to micturition. And this whole process is known as micturition reflex. Are these points clear? Now urine is slightly acidic, pH should be around 6. And how much urea do you release every day? 25 to 30 gram. Clear? The presence of presence of glucose is glycosuria, presence of ketone bodies is ketonuria. And we have already read that the presence of these two substances, they indicate diabetes mellitus. Is that clear? So ketonuria and glycosuria, or you can call it glucosuria, they are indicative of diabetes mellitus, sugar, right? There are other organs in excretion, involved in excretion. Lungs, they excrete carbon dioxide. How much? 200 ml per minute. And water. Liver secretes bile containing substances like bilirubin, biliverdin, cholesterol, steroid hormones, vitamins and drugs. Then your sweat glands secrete, they release your water, sodium ions, some urea and even lactic acid. Sebaceous glands are the oil glands. What do they excrete? Sterols, hydrocarbons and waxes. That is why oil is waxy, right? And then some nitrogen, saliva. Your saliva also excretes certain things. What small amounts of nitrogen is waste. So do you know even your saliva has slight amounts of urea? Is it clear? Now coming on to the disorders. So see, of course, our urine is going to excrete urea, but there is some amount of urea always present in your blood, but it should be under a certain, it should be within a certain limit. Okay. Now, if the level of urea in blood increases, that is known as uremia. So in that case, it means the kidney is not functioning properly. So either we'll have to go for artificial kidney, that is hemodialysis, or we'll have to get a kidney replaced, that is kidney transplantation. Let's first talk about hemodialysis or the artificial kidney because it functions like kidney. Now what happens? Blood is taken from convenient artery. We add a anticoagulant in it, that is heparin. Otherwise, as soon as the blood comes out, it will start coagulating. Now, this blood with anticoagulant is passed through the cellophane tubes. Now, the cellophane tubes are surrounded by a dialysing fluid. Now, this line is very, very important, that the dialysing fluid has the same composition as that of plasma, except that there is no nitrogenous waste present in the dialysing fluid. But your plasma has nitrogenous fluids. Are these points clear? Now, cellophane membrane allows the passage of molecules on the concentration gradient. See, we are passing the blood of that individual whose kidney is not functioning. So that person has high amount of urea in his blood. But there is no urea in the dialysing fluid. So what will happen? Urea will come out from the cellophane membrane. It is a selectively permeable membrane, right? From high to low concentration, and that is how our blood will become nitrogenous waste free. Is this clear? And so, but we have to keep changing the dialysing fluid, otherwise this urea can pump, it can go back into your blood also. So once the blood is clear of urea, we pump it back to the body by adding anti-heparin now, so that when needed the blood can coagulate once it is inside. Is this clear? We'll take a 2-minute break. Give me 1 minute to complete this chapter now. So once, see, when there is a chronic problem, when there is an acute renal problem, and ultimately it has to be solved by getting a renal transplant or a kidney transplant. Is that clear? So acute renal failures have to be solved by kidney transplantation from a close relative to decrease the chances of rejection. Are these clear? Then what is renal calculi? Calculi word is for stone. So any stone or insoluble mass of crystallized salts in the kidney are called renal stones. Now these salts are majorly of oxalates. Is that clear? And then glomerulonephritis. Itis ayato, it is going to be inflammation. Inflammation of what? Glomeruli. Are these points clear? All of you give me a green heart or a thumbs up that you are thorough with all these chapters. You've read all these chapters with me. Quickly let me know. Quickly let me know. All these clear? Please let me know. Yes, please. Let me know that these chapters are super, super clear. And now we are going to start with another chapter of human physiology, that is body fluids and circulation. Thank you all. I am reading the chat along. Take a break, take a 1 minute break, and in that 1 minute break, you're going to send me lots of hearts, and you're going to like the channel, you're going to subscribe to the channel, and remember, tomorrow, guys, at the same time, that is 7 p.m., we are going to start with complete class 12 revision. We are going to start with complete class 12 revision. Is this clear? Okay, I'll take a doubt in this 1 minute break. What is RAS mechanism? Afan is asking. RAS stands for, see, it is an abbreviation. R for renin, angiotensin, aldosterone. So basically, when your blood pressure decreases, this RAS mechanism gets initiated. Okay, your kidney will send renin. The kidney has juxtaglomerular cells. It is going to send renin. Renin will enter into the blood, and your blood already has angiotensinogen, right? So renin is going to convert angiotensinogen into angiotensin 1. Later, this 1 will be changing into angiotensin 2. Now, this angiotensin 2 is the main hero. This angiotensin 2 is going to promote vasoconstriction. And when vessel will contract, what will happen? Area decreases, pressure increases, blood pressure increases. Secondly, it is going to promote the secretion of aldosterone, sodium reabsorption, more sodium, more water reabsorption, blood volume increases, blood pressure increases. Is this clear? All right. Break is over. Guys, I am telling you once again, those of you who are finding some difficult topics during the session, some topics difficult during the session, see, this is a clear indication that you need a detailed explanation there. Please try not try, do that. Please make a list of those topics, those small topics which you are finding difficult. Clear? So make a list. So as I've already informed, your unit wise revision will continue from Monday onwards, right? But why I decided to take this class 11 and 12 revision right now, right away? Because from tomorrow onwards, your mock test series, free mock test series is starting. So make sure that you attend, you take those mock test series. So we are not selling anything, guys. This is for you. It is free, right? It is absolutely free starting tomorrow from 2 to 5 p.m. You have to take it. That will actually give you an insight. I'll tell you why it is important. It will give you an insight into how much you are well prepared. And now, after taking that mock test, when you'll come back to the unit wise revision, you will actually know what to pick from the session. What are your doubts? You can actually ask your doubts during the chat. I mean, during the live session, right? Ma'am, ਤੁਸੀਂ ਕਿੱਥੋਂ ਹੋ? I am from Punjab. All right. I have missed my dinner today. So can you also wait? All right. All right, guys. Moving on, moving on. Let's take a minute break. Coming back, coming back. All right, guys. I'm back. The break is over. Yes, it was a very, very short break. Now, coming on to the chapter 15, that is body fluids and circulation. See, we have already read about circulatory system in various phylums of animal kingdom. Sponges and coelenterate, they show circulation through body cavities, right? Then blood is the most commonly used fluid. Also the lymph, it represents a special connective tissue. Then blood has two components, plasma and the formed elements. Formed elements are the cells. Quickly keep revising. If you have missed on any important line you have not highlighted or you forget that, please put a star mark or a hashtag and keep making a list of the difficult topics that you're finding during the chapter. Okay? Yes. Now, plasma is straw colored, constitutes 55%. How much water is present in plasma? 90 to 92%. And 6 to 8% are the proteins. What are the three different types of major proteins present in blood? Fibrinogen, globulins, and albumins. Now, fibrinogens are for clotting and coagulation of the blood. Globulins are for defense mechanism. Albumins help in osmotic balance. Is this clear? Then the factors, clotting factors are also present in blood plasma. Now, if we remove these clotting factors from plasma, the remaining part is known as serum. So plasma without clotting factors is known as serum. Are these points clear? All right, moving next to the formed elements. Formed elements are of three types: RBCs, WBCs, and platelets. RBCs are the most abundant. They don't have a nucleus, biconcave in size. They have a protein known as hemoglobin, which is actually going to help in gaseous transport, right? How much hemoglobin do you have? Remember, it is in grams. Sometimes we make a mistake for the unit. 12 to 16 grams of hemoglobin in 100 ml of blood. What is the lifespan? 120 days. Now, where do these RBCs get destroyed after 120 days? So, they are buried in spleen. So, it is known as the graveyard of RBCs. Are these points clear? Talking about the second type of formed element, that is leukocyte or WBC, nucleated, lesser, in fact, the least in number, only 6,000 to 8,000 per millimeter cube, generally short lived. Is that clear? Now, there are 5 different types of WBCs. You can see this in the diagram also. So, the most abundant are the neutrophils. How much? 60 to 65%. You can get this question and match the following to match the different percentage. So, you must remember. See, a simple trick I am giving you how to remember. You have to remember null map. You have to remember null map. Null, N for a neutrophil, L for lymphocyte, M for monocyte, then A for acidophil and B for basophil. So neutrophils are the most abundant, basophils are the least abundant. Neutrophils and monocytes monocytes, they constitute 6 to 8%, and both neutrophils and monocytes are phagocytic in nature. Basophils are secretory in nature. What do they secrete? Histamine, serotonin and heparin, right? And they are least, how much? 0.1%, hardly 0.1%. Clear? Then eosinophils are 2 to 3%, and they have important role to play during the allergic reactions. Is it clear? Lymphocytes, see written 20 to 25%. There are two types. Now depends where do they get specialized. If they are specialized in the bone marrow, we call them B lymphocyte. If they are specialized in thymus, we call them T lymphocytes. Third type of formed element is a platelet called the thrombocytes. Thrombocytes are not real cells. They are actually just cell fragments. They are sticky in nature. They are cell fragments. And the master cell which produces the platelets is known as the megakaryocyte. It's known as the megakaryocyte. Are these points clear? Now, where are all these type of cells formed? They're all RBC, WBC platelets, all are formed in the bone marrow. Now, what is the number of platelets in your blood? It is 1.5 lakh to 3.5 lakh per millimeter cube. So, the most abundant RBCs, second abundant are platelets, and the third one are the WBCs. Let me have water. Okay. Moving on now. So what is the function of platelets, guys? The function of platelets is the blood coagulation or clotting. Is the blood coagulation or clotting. So any reduction in the number of platelets will lead to excessive loss of blood. Now we know that bloods are grouped into some categories. So based on which, so we have different types of blood groups, right? Now, these different types of blood groups are on the basis of some special proteins present on RBC. We call them antigens. See, the presence or absence of two surface antigens on RBC. Where are these antigens? On RBC. So, in response to these antigens, we have antibodies present in your plasma. Is that clear? So see, we have four types of blood groups: A, B, ABO and O. So the blood group depends upon the antigen. So antigen on RBC and the blood group is exactly the same. Do you see here? But antibodies are just the opposite. So if we have A antigen, antibody is for B, anti-B. B antigen, B blood group, but antibody for anti-A. When blood group is AB, it means we have both types of antigens, but there are no antibodies, and that's what makes them universal recipients. And O blood group, no antigen, neither neither A nor B, but they will have both type of antibodies in their plasma. So that is why, see, since they have no antigens, this makes them universal donors. So this is assertion reason. Why are O universal donors? Because there are no antigens. Why are AB are universal recipients? Because they have no antibodies. Are these points clear? Clear to all? Now we have another kind of antigen present on the RBC surface, which is RH. Apart from A and B, we can have another kind of antigen. Rather, there are so many antigens present, but we just need these two for proper and successful blood transfusion. Now this is RH. RH word comes from rhesus monkey, from where it was first discovered. Now, individuals which have RH on their RBC are RH positive. Those who don't have this antigen are known as RH negative. Clear? Now, remember, this RH is going to play a very crucial role during pregnancy. So, see, if mother is RH negative, but the baby is RH positive. Why? Because the husband was RH positive. Out of positive negative, it is a positive that dominates. If one of the partner is positive, the bacha is going to be positive. Clear? Now see, if mother is RH negative and father was RH positive, it can lead to this condition known as erythroblastosis fetalis. When? Why? See, RH antigens on fetus don't get exposed to the RH negative blood of mother in the first pregnancy. First pregnancy is always normal. Why? Because the two bloods are separated. But during the childbirth, if there is exposure of RH antigen to the mother's body, to the mother's blood, then mother's body is going to start preparing these RH antigens, antibodies. So mother is going to start preparing these antibodies against the RH antigen. Now, first pregnancy has happened, first birth has happened, all safe and successful, all happy. But in the subsequent pregnancies, what might happen? The antibodies which the mother has produced, now they might enter into the fetus and kill the fetus. They will destroy the fetal RBCs. Now, excessive destruction of RBC fetus will lead to jaundice and severe anemia. Now, this will lead to the death of the baby. This condition is known as erythroblastosis fetalis or hemolytic disease of newborn. But these days, so in such cases, what happens? Only first pregnancy is successful. Rest all the subsequent frequencies will lead to stillbirth. But nowadays, it is not really a problem. If wife is Rh negative and husband is Rh positive, still we can have as many successful pregnancies as possible. But in that case, mother has to be administered with anti-Rh antibody immediately after the delivery of the first child. Are these points clear? Now coming on to the coagulation of blood. Clot, or also known as coagulum. How is clot formed? By the network of threads called fibrils. So basically what is written here is in the backward style. So it starts with actually release of thromboplastin. Okay, so whenever there is an injury, your platelets release a thrombokinase. Your platelets release this enzyme called thrombokinase. This thrombokinase converts prothrombin into thrombin. Thrombin will convert inactive fibrinogen into fibrins, and fibrins will trap the blood vessels to form a proper clot. Are these points clear? Are these points clear to everyone? So yes, all physiology, not only physiology, we are going to complete entire biology. We still have 2 hours with us. 2 hours are gone. We still have 2 hours. Human physiology is taking longer time. Rest of it will not take as much time. Is this clear? Now see, calcium ions play a very important role in clotting. And guys, since now you are all here, let me tell you 2 very important things. Tomorrow at 2 p.m. to 5 p.m. Starting tomorrow, you have to take this free mock test series. And then these series will be so useful, that unit-wise revision will be so useful. Side by side, you start discovering your weakness. You start getting acquainted to the NEET exam, that practice of real NEET exam. Okay, and start making a list also. And tomorrow we are going to complete class 12 at 7 p.m. Is this clear? All right, moving on to lymph. This is the second type of fluid connective tissue present. Now, the fluid which is released outside the cell, which is present outside the cell, is known as interstitial fluid or the tissue fluid. It helps in the exchange of nutrients and gases. Lymph is colorless while blood is red. Why is lymph colorless? Because it does not have hemoglobin. It is known as lymph because it has maximum lymphocytes, and lymphocytes, as we know, are responsible for the immune response. Is this clear? They are important carriers for nutrients, hormones. Fats are absorbed also through lymph in lacteals. Now, what are lacteals? Lacteals are the special lymph vessels present in the intestinal villi. Your intestine has many many finger-like processes to increase the surface area for absorption. So, beneath every intestinal villi is present a lymph capillary. This lymph capillary is known as a lacteal. Is this clear? All right. Moving on, guys. So all those of you who have joined me later, let me tell you, I have started from backward. I've done endocrine, I've done nervous system, I have completed movement and locomotion, excretory, and now we are discussing circulation. Okay, one more chapter of human physiology is left. We are going backward to fourth. Is that clear? Okay. Now, moving on to circulatory pathways. There are 2 types of circulatory pathways we have already discussed in animal kingdom: open and closed. Open is in arthropods and mollusks, and closed is in annelids and chordates, right? Now, all vertebrates we know have a muscular chambered heart. We have read that fishes have 2-chambered, amphibians and reptiles have 3-chambered, except crocodiles. So, crocodiles are in our category. Crocodiles, birds and mammals have 4-chambered heart. Is that clear? Fishes have single circulation. Amphibians and reptiles show double circulation, but because of mixing of blood, we call it incomplete double circulation. While in birds and mammals, there is complete double circulation. Blood enters the heart twice, once in the form of oxygenated and in the deoxygenated. and the two bloods are completely separated. That is why we call it complete double circulation. Are these points clear to all? Then moving on to the human circulatory system, we have heart. So there are three components of human circulatory system. What? Heart, the blood vessels, and the blood. So we have read about blood already. We are talking about heart now. What is the origin of heart? Overall, your circulatory system is mesodermal in origin, and so is your heart. Where is heart present? In between the two lungs, slightly tilted towards the left, and that is why you feel your heartbeat towards the left region. Now, heart also requires a protective covering. Now, the protective covering of brain. Quickly, let me know what is the protective covering of brain. And then we've also talked about the protective covering of kidney. Let me know these two names in the chat box. Quick, quick, quick. And guys, there is a link given in the chat already. You can enroll for the All India Mock Test Series absolutely free. The link is pinned in the chat. Okay? Yeah. Hi, everyone. All those of you who have joined me late, please go with the flow and then you can watch the chapters that you have missed later on. Yes. So the protective covering is known as pericardium. Right. Right, Shri. Right, Shri. The protective covering of brain was meninges and the protective covering of kidney was renal capsule. Now talking about heart, it is a pericardium and it is double layered. So there is a fluid in between, pericardial fluid. We know there are atria, ventricles, right? We know this. And then right atria, right ventricle has bicuspid valve, which is which it has tricuspid valve, which has three flaps. And then left auricle and left ventricle has two flaps, which is bicuspid valve. Are these clear? So on the right side we have tri, on the left side we have bicuspid. This is the structure of the heart. This is the structure of the heart. Now, what is the function of valves? They always prevent backflow. The entire heart is made up of cardiac muscles. We have talked about cardiac muscles enough times, right? Now see, very important. This specialized cardiac musculature is called the nodal tissue. Okay, there is a specialized cardiac musculature known as the nodal tissue present in the upper right corner of the right atrium, which is known as the sino-auricular node. Now it is the sino-auricular node at which the impulse will be generated, which is going to generate the heartbeat. Apart from SAN, that is sino-auricular node, we have AVN, atrioventricular node. Where is this present? In the lower left corner. So both SAN and AVN are in the right auricle. Please understand, both SAN and AVN are in the right atrium. SAN in the upper right corner, AVN in the lower left corner. Is it clear? Lower left corner of the right atrium. Then after SAN, impulse goes to AVN. After AVN, it goes to a bundle of nerves known as AV bundle, which enters the atrioventricular septum. And then AV bundle then divides into small fibers known as Purkinje fibers. So nodal musculature, that means the SAN, has the ability to generate the action potential, that means the impulse. So automatically, it is pre-designed that nodal musculature will produce impulses. So that is why it is known as auto-excitable. No external stimulus is needed to excite it. It will be excited automatically, auto-excitable. And how many times will it produce these impulses? Maximum of 70 to 75 times per minute. This is a normal time. Okay. So as many times SAN produces impulse, that many times your heart beats. So the number of impulses produced by SAN is exactly equal to the heartbeats. Is this clear? And that is why SAN is known as the pacemaker. Now there are four chambers. Talking about the cardiac cycle, the mechanism of heartbeat. See, heartbeat is understood by cardiac cycle. There are four chambers of heart. Now, first of all these four chambers are in the relaxed state. So when all are relaxed, we call it joint diastole. Now understand what happens during the joint diastole. During joint diastole, your TV, that is tricuspid valve, and BV, that is bicuspid valve, both TV and BV are open. So when TV and BV are open and your heart is in the relaxed state, there is negative pressure in the heart. So blood from the pulmonary veins and vena cava is going to flow into the ventricles. So right side has deoxygenated, left side welcomes the oxygenated blood. Is that clear? Now at this time, SLV, that is semilunar valves, are all closed. So all these red lines show your joint diastole. Now what happens? This joint diastole, by the way, goes for 0.4 seconds, 0.4 seconds. Okay. Now, at the end of 0.4 seconds, SAN suddenly generates an action potential. So when an impulse is generated by SAN, it is spread to both the atria and both the atria start contracting. Now, atrial contraction is known as atrial systole. Now, what will happen? The blood which is flowing towards the atria, when atria are contracting, that blood will forcefully be sent into the ventricles. Now, rest of the, see, 70% ventricles were filled during the joint diastole, 30% will be filled during the atrial systole. Is it clear? Then uh, it just goes for 0.1 second. Now at the end of 0.1 second, by this time, see, impulse has already reached the ventricles. Now ventricles will start contracting, known as ventricular systole. Now atria will undergo relaxation. Is that clear? So ventricular systole is equal to atrial diastole. Is that clear? Now, it is overlapped by atrial diastole. Now, what happens first? During the ventricular systole, the first thing is closure of TV and BV. Now, as soon as tricuspid and bicuspid close, the first heart sound is produced. Now, the pressure increases within the ventricles. This leads to the opening of semilunar valves. See, ventricular pressure increases, semilunar valves open. Now, the blood is going to flow out through pulmonary artery into the lungs and aorta to the body. Now, when the blood goes out, now ventricles start relaxing. It is the end of that systole. Now, during the towards the end of the diastole, sorry, yeah, towards the end of the diastole, or you can say it's almost there to relax, the ventricles are going to close back, producing the second sound. Now, when the ventricles, these SLV close, the TV are going to open. The TV are going to open. Ventricles and atria now all in the relaxed state once again. So this is known as joint diastole. Is this clear? Now sequential event in the heart which is cyclically repeated. This sequence goes on repeating again and again, and this sequential event is known as cardiac cycle. So overall, between two heartbeats, the time period is known as, or the events are known as, the cardiac cycle. So cardiac cycle, the number of cardiac cycle is equal to heartbeats. So there are three things. The number of impulses produced by SAN is equal to the heartbeat and is equal to the number of cardiac cycles. Is it clear? These three things are always equal. Now the total duration of cardiac cycle is 0.8 seconds. Now how much blood is pumped out during each cardiac cycle? That is 70 ml, and this is known as stroke volume. Now if we multiply stroke volume with the heart rate, that gives us the cardiac output. So cardiac output is total volume pumped out by heart in 1 minute, which is actually the total amount of blood, which is 5000 ml or 5 liters. Now the body can alter stroke volume also, body can alter your heartbeat also. So overall your cardiac output can change from time to time. Is that clear? Now very important, cardiac output of athlete is much higher than the ordinary man. Now, how do you measure how do you listen to the heart sounds, or how do you measure your heartbeat? It is through this device known as stethoscope. The two sounds, major sounds, first sound is lub during the closure of TV and BV, and second sound is dub during the closure of SLV. And these sounds have clinical diagnostic significance. Now, very important aspect is ECG. First of all, please see the full form. ECG stands for electrocardiogram. Not graph, not graphy. ECG is electrocardiogram. This is according to the new NCERT. Now the machine used for this ECG is electrocardiograph. So what is ECG? Machine is graph, and the graphical representation is known as electrocardiogram, which shows the electrical activity of the heart. Is it clear? Now in order to measure, we need to fix 3 electrical leads. One on each wrist and one to the left ankle. At least these 3 leads have to be pasted on the body. Other than these, the number of leads can be increased to 12 as per the need as well. Is that clear? But at least on the minimum side, there has to be 3 leads. Is that clear? Now, see, this is the diagram. You should know the labelings. This is P, Q, R, S, and T. Now, what does the P wave represent? P wave is the excitation of atria. Now, Excitation or depolarization of atria is not same as contraction. It is excitation. Contraction is going to take place later on, and moreover, contraction is the mechanical change. What ECG tells us is about the electrical activity, not the mechanical activity. Are these points clear? Right. Now, QRS complex, this represents depolarization of ventricles. And see, it is clearly written, contraction will start shortly after Q. Clear? And then T wave represents the return of ventricles from excited to normal state, that is repolarization. End of T wave marks the end of the systole. Is this clear? Now, if you have to count the number of heartbeats, are you going to like measure all the waves from this P, this is Q, this is R, this is no. The simplest way is you just measure the number of QRS complexes. Number of QR complexes is equal to the number of heartbeat. So now we have four things. See, number of heartbeat is equal to number of impulses produced by the SAN is equal to number of cardiac cycles is equal to number of QRS complexes. Are these points clear? Yes. Is this point clear? All right. So any deviation in the shape indicates abnormality and has clinical significance. Now coming on to the double circulation, we understand double circulation is the blood is going to enter the heart twice. So third component of our system is, of our circulatory system is the blood vessels. There are three types of blood vessels, arteries, veins and capillaries. The two main ones, artery and vein, is being discussed. See, both artery and vein has three layers in the wall. The inner lining is squamous epithelium. The middle one is tunica. Yes, the middle one is tunica intima, the middle layer, and it is made up of the inner layer is known as tunica intima, which is made up of squamous epithelium. The middle layer is known as tunica media, made up of smooth muscles and elastic fibers, which will allow dilation and constriction, right? And the outermost layer is known as tunica externa, which is made up of fibrous connective tissue or collagen fibers. Clear? Now, the basic difference between artery and vein comes from tunica media. In artery, this media is thicker. In vein, it is thinner. Is that clear? And eventually, and that is why the area of the lumen also changes. Now, since we are talking about double circulation, there are two circulatory pathways. One is pulmonary circulation, that is between heart and lungs, and then there is systemic, that is between heart and the body. Is this clear? And we have one special circulatory system, that is between two organs, connecting two different organs and then the heart. We call it portal circulation. Like from all the blood from your digestive tract goes to your liver first and then to the heart. So this connection between digestive tract and liver is known as hepatic portal system, carries blood from intestine to liver and then to the systemic system. And then the fourth system is the coronary system, that is a special circulation between heart and the muscles of the heart. Are these points clear to all? Please give me a thumbs up that all things are clear till now. Quickly let me know. Yes, please. Are you revising all things? Guys, it is practically impossible for all of you. I can definitely challenge. It is practically impossible for you to read NCERT in 4 to 5 hours without these kind of sessions. Because on our own, we don't have that rush, and honestly, we all tend to miss on some keywords. We all tend to miss on these keywords. But when we are reading together, we can read it within that small time period and we can't miss on any important line. And these lines which I have underlined, they already include PYQs. Is this clear? Yes. Now, regulation of the cardiac activity. Normal activity is regulated intrinsically. Why? Because we know that your heart is auto-excitable and myogenic, right? But medulla oblongata can moderate the cardiac activity through autonomic nervous system. Now, sympathetic nerves, we know, will increase the heartbeat, the strength of ventricular contraction and the cardiac output, but parasympathetic is going to do the opposite. Whereas adrenal medullary hormones, both adrenaline and noradrenaline, they are both going to increase the cardiac output. Are these points clear? Are these points clear to all? So all those of you who have joined me just now, guys, we are doing NCERT reading of class 11. We are going to complete class 11 today. I mean tonight, right? And I am almost done with human physiology. We have started from backwards. Those of you who have missed the previous chapters, you can do that later in the recorded session. But you can just go with the flow. Don't miss on these chapters now. Since you have missed the previous chapters, doesn't mean that you can't carry on now. You can start right away. You can start right away. I'm going to start with the last chapter of human physiology after these disorders. So just stay with me. Now, disorders of the circulatory system. See here, high blood pressure, also known as hypertension. The indication of high blood pressure is your systolic pressure is 140 and your diastolic pressure is 90. So 140 or 90 represents your hypertension. Then coronary artery disease. By the way, high blood pressure will have side effects on on your heart health, and it will affect structures like brain and kidney. Then coronary artery disease, that is CAD, also known as atherosclerosis. Now it is due to deposition of calcium, fat, cholesterol and fibrous tissue, which makes the lumens of arteries narrow. Then we have angina. Angina also known as angina pectoris because it affects the pectoral muscles here. Okay? Now there is severe chest pain. Why? Because of no insufficient oxygen supply to the heart muscle. It can occur in any age, more common in middle-aged and elderly, and due to less blood flow. Then there is another condition, heart failure. Heart failure means blood has failed to pump enough blood. And then it can also occur due to congestion of lungs. So we also call it congestive heart failure. Then we have last disease, that is cardiac arrest. Cardiac arrest means heart has stopped beating altogether. There is sudden collapse of the person. And last not the least is heart attack. Heart attack means when heart muscle is suddenly damaged by inadequate blood supply. Is this clear? Inadequate blood supply. Please let me know that all things are super clear. Give me a thumbs up that you are done with this chapter with me. You're done with this chapter with me. Quickly let me know. And we are going to start with the last chapter of human physiology, guys, breathing and exchange of gases. Give me a thumbs up that you all done this chapter with me. Yes, please. It's time for the thumbs up. All clear? Okay. Moving on to the last chapter of human physio, that is breathing and exchange of gases. So there are two separate terms, breathing and respiration. See, oxygen is utilized to indirectly break down simple molecules of glucose, amino acids, fatty acids to derive energy. This is respiration. And exchange of oxygen from atmosphere and CO2 with the cells, with CO2 produced by the cells. This is known as breathing. Is that clear? Then respiratory organs, we already know, we have discussed it in animal kingdom. Coming directly to human respiratory system, starts with the external nostrils, going on to the nasal passage, then the nasal chamber, going into the pharynx. Then pharynx leads to the voice box known as larynx, then trachea. Now voice box is basically a cartilaginous box, and since it produces sound, it is known as the voice box or the sound box. Clear? Now the opening of glottis. Glottis is the opening of larynx. Okay. Now opening of glottis is opening of larynx, that is glottis, is covered by a thin elastic cartilaginous flap called epiglottis. What is the function of epiglottis? So whenever we have to swallow, epiglottis shuts down the passage of larynx so that food doesn't enter into the larynx. Now trachea, remember, trachea is the windpipe. It is going to divide into two bronchi. At which level? Fifth thoracic vertebra level. Is that clear? So we have two bronchi, and these bronchi are going to enter each lung. Each bronchi will further divide into secondary and tertiary bronchi, and then into bronchioles, and finally into terminal bronchioles. And these bronchioles will end up into vascularized bags known as alveoli. Now, throughout this passage, starting from trachea, going up to, see here, this green colored line, trachea, going up to the initial bronchioles. Remember, initial bronchioles, they have these incomplete cartilaginous rings, C-shaped cartilaginous rings. Are these points clear to all? Then protective covering of lungs is the pleura. Again, double membraned, just like pericardium. And there is a pleural fluid also. This is the structure of your respiratory system. Now, the whole respiratory tract is divided into two parts. Conducting part and the respiratory part. Is that clear? So external nostrils to terminal bronchioles. This is known as the conducting part. Very important. This is a PYQ. Please focus. And then alveoli along with the alveolar ducts. Their ducts means alveolar ducts. They constitute your respiratory or exchange part. So you should be very clear about this distinction. So nostrils to bronchioles, terminal bronchioles is the conducting part. It only conducts the gases. But the alveoli and the ducts, they actually help in exchange of these gases between blood and the alveoli, right? So that is why we call it respiratory part. So what is the function of conducting part? Only to conduct. While not only to conduct, apart from conduction, it also clears the foreign particles. So there are hair, there is mucus lining the tract. So it clears the foreign particles. It humidifies, it adds moisture. It also brings the air to the normal body temperature. So thermoregulation is also the part of function of conducting part. While exchange part is only involved in actual diffusion of gases. Are these points clear? Now, lungs are present in airtight chamber known as the thoracic chamber, and thoracic chamber is formed by vertebral column, sternum, and your ribs. And on the inferior side, on the lower side, we have dome-shaped diaphragm. Now, respiration is going to take place in 5 steps. First, starting with exchange between atmosphere and lungs. This is known as breathing or pulmonary ventilation. Then, diffusion of gases across the alveolar membrane. And then, third, transport of gases by blood. Fourth is diffusion between blood and tissues. And fifth, utilization of oxygen by the cells to actually produce energy. This is called cellular respiration. Are these points clear? Then what is inspiration? Inspiration is when the air goes in. How will it take place? There is simple basic principle of physics. Air moves from high pressure to low pressure area. So if we want air to be inhaled, there has to be low pressure within the lungs. So see, inspiration can occur if pressure within the lungs, that means intrapulmonary pressure, is less than the atmospheric pressure. So negative pressure has to develop within the lungs. And then expiration, air goes out. When will this happen? This will happen when the atmospheric pressure will be lesser and intrapulmonary pressure will increase. Now, this maneuvering of the pressure will can occur because of certain structures like diaphragm, like your ribs. Now, see, it is clearly written, contraction of diaphragm increases the volume in the anterior-posterior axis. Clear? While contraction of the external intercostal muscle lifts the lips and sternum and increases the volume in the dorsoventral axis. So this diagram is very, very important, and it has come for in your PYQ. Clear? Now increase in the intrapulmonary pressure. So decrease in the intrapulmonary pressure will lead to inhalation. Yes, and the reverse is true for expiration. Now see, there, how many times do you breathe in 1 minute? It is 12 to 16 times. So this is your breathing rate. Don't get confused with the heart rate. Heart rate is 72 to 75 or 80 times per minute. But your breathing rate is 12 to 16 times per minute. Are these points clear to everyone? Are these points clear to everyone? Volume of air involved in breathing movements can be estimated. So how much volume of air are you inhaling or exhaling during a special kind of during a specific type of breathing? That can be measured by spirometer, and this helps in accessing the pulmonary functions. Now this topic is very, very important, respiratory volumes and capacities. There is not even a single paper, I guess, not even a single year has passed by without getting question from this chapter, this topic. Such an important topic. Let's take a break. 1 minute. Let me take a break. I want to know from all of you. I want everybody to chat for a while. Let me know how many chapters have you discussed with me or attended with me in this session. I want all of you to write down in the chat. This is the last chapter of Human Physiology. How many of you have attended all the chapters, or however chapters with me in this session? Shanu says 0. Then what are you doing, Shanu? Avantika has done 2. Dr. Paila has done 5. Okay. So we have done endocrine, nervous, movement, excretory, then circulatory, and this is the 6th one. So you have missed 1, 3, 4, 2, 5. I've attended 5 chapters. 5, 5, 5. Okay. Okay, 5. Don't spam. 5 is enough. Once. 2, 3. I'm from starting. Very good. Very good, Dikanta. Okay, Shanu. 5, 1. From starting. Very good. Complete human physiology done. Very good, Yuvraj. All right. All till now, Prateek. Perfect. What about others? Quickly let me know. 0. Why, Sharad? Why 0? What are you doing then? Wasting your time? So, guys, coming back to the topic, volumes, tidal volume. See the keywords here. Very good. Very good. All those of you who have joined me since the beginning of the session, excellent. In case you could not because of the other session going on parallel, you can watch those chapters later on. But those of you who have joined me right now, please go with the flow. Okay, yes. Tidal volume, guys. Tidal volume is a volume inspired or expired during normal inspiration. And what is the value? 500 ml. So 500 ml per breath. So how much in 1 minute? That would be 500 into 12. That is 6,000, or into 16. That would be 8,000 milliliter of air per minute. Or you can say 6 to 8 liter per minute. Inspiratory reserve volume is forcible inspiration, inhalation. See, person can inspire forcible inspiration. So volume of air during inhale, forcible inspiration that you take, that is 2,500 to 3,000 ml. So this is apart from the tidal volume. Remember that. IRV is above tidal volume. So apart from 500 that you usually take, you can take extra of 2,500 ml to 3,000 ml. But in IRV, your expiration is normal. Now similarly, in expiratory reserve volume, expiration is forcible, but inspiration is normal. So how much extra can you exhale? 1,000 to 1,100 ml. Now residual volume. So no matter how forcibly do you breathe, there is certain amount of air which will always be left behind in the lungs, and that is known as the residual volume, and this averages between 1,100 ml to 1,200 ml. This is residual even after forceful expiration. Is that clear? Now, what are the capacities? See, remember, when we are discussing volumes, they are always single entities here. But in capacities, we have more than one entity. Like, for example, inspiratory capacity is TV plus IRV. Expiratory capacity is EC. That is TV plus ERV. What is functional residual capacity? ERV plus RV. So when we exhale forcibly, and still some volume is left inside, that is residual volume. But see, we are not always respiring forcibly. We are not always exhaling forcibly. So when we are exhaling normally, the volume of air which remains behind at that time after normal expiration, that is FRC. So I hope you understand the difference between FRC and RV. Now, vital capacity is a combination of three things: ERV, TV, and IRV. So basically, in vital capacity, expiration and inspiration both has to occur to the maximum or forcibly. So forcible expiration followed by forcible inspiration, or forcible inspiration followed by forcible expiration, that is vital capacity. Are these points clear? Are these points clear to all? And then we have TLC. TLC is all elements combined together: RV, ERV, TRV, and IRV. Is that clear? Or you can simply write VC plus RV. Now you can get numericals from this topic, or you can just get the values to be matched. Usually NTL likes to give match the following, so get ready with the values. Is that clear? Moving on to the exchange of gases. So primary site for the exchange is the alveoli. Simple diffusion. Now diffusion will be affected by the thickness of the membranes. Now these are the partial pressures. See, this whole exchange is a game of partial pressure difference, and every gas is going to move independently of the other, and from higher pressure area to lower pressure area. Is that clear? So you must know the values written in the table. Then moving on to the solubility. So solubility of carbon dioxide is 20 to 25 times higher than oxygen. Very important value, very important data. Now the diffusion membrane, see here, the diffusion membrane is formed by endothelium membrane of the blood capillary, followed by the basement membrane, and then the and the squamous epithelium of the alveolar wall. Is that clear? So there are three layers: endothelium of blood capillary, basement membrane, and the squamous layer of the alveoli. These three together constitute the diffusion membrane. And what is the total thickness? Much less than 1 millimeter. So this is how thin the diffusion membrane is, and that is why diffusion gets possible. Is this point clear? Then transport, very important. For oxygen, 97% of oxygen is transported by hemoglobin forming oxyhemoglobin. 3% goes through the plasma. For carbon dioxide, 20 to 25% goes through hemoglobin in the form of aminohemoglobin, 70% in the form of bicarbonate ions, and 7% in the form of plasma. Now let me know in the chat box how much total percentage of carbon dioxide is transported through plasma. Yes, please. If you can answer that, your NEET is definitely clear. Definitely clear. Now, talking about the transport of oxygen. So, major chunk of oxygen, that is 97%, is going to be transported by RBCs, which has this respiratory pigment called hemoglobin, red in color, binds with oxygen reversibly. One hemoglobin can carry maximum of 4 oxygen molecules. Now, this can be studied through this ODC, oxygen dissociation curve. Now, see this graph. It is a sigmoid shaped graph. Now, on the on the x-axis, we are taking partial pressure, and on the y-axis, we are taking percentage saturation. You have to observe all these things. These are all PYQs. Is that clear? Now, okay. Now, this ODC can shift towards the right and towards the left. Now, these right and left shifts will show the tendency. They will be affected by many factors. See, whenever this graph shifts towards the right hand side, it means oxygen and hemoglobin association is decreasing. It represents oxygen hemoglobin dissociation. While the this left hand shift is going to show oxygen hemoglobin association. In alveoli, see alveoli, the conditions like, which are the conditions in alveoli? Alveoli is high pressure, partial pressure of oxygen, low partial pressure of CO2, very important aspect, lesser H positive concentration and low temperature. All in alveoli, all these factors are favorable for the formation of oxyhemoglobin. While in tissues where there is more of CO2 and less of oxygen, high of H positive and higher temperature, these factors, they will favor the dissociation and curve will take the right hand shift. Is it clear? So every 100 ml of 100 ml of blood can transport or deliver 5 ml of oxygen, and every 100 ml of blood can transport only 4 ml of carbon dioxide. Are these points clear? Now, for carbon dioxide, the major chunk is transported in the form of bicarbonate ions. Please see this, in the form of bicarbonate ions. How? See this reaction. Carbon dioxide plus water in the presence of this fastest enzyme, carbonic anhydrase, forms carbonic acid, which further dissociates to form bicarbonate ions and H positive ions. Now, this reaction is occurring in RBCs as well as in plasma. But plasma has, it is clearly written, this enzyme, the minute quantities of the enzyme are present in plasma too. So this reaction is occurring in both plasma and in RBC. But because RBC has more Ca, carbonic anhydrase, so more of the bicarbonates will be produced within the RBC. Are these points clear? Now talking about the regulation. See, medulla region of brain, that is medulla oblongata, has respiratory rhythm center. Then pons varolai has pneumotaxic center. Pneumotaxic center, as many signals will, the signals which will be generated by pneumotaxic center will reduce the duration. So as long as pneumotaxic center keeps on generating signals, the duration of inspiration goes on decreasing. Then guys, we have a chemosensitive area. This chemosensitive area, as the name says, is sensitive to chemical. Which two chemicals? Carbon dioxide and H positive ions. Now remember, these sensitive areas are present, these receptors are present in the aortic arch, that is your aorta, and carotid artery, which goes to your brain, right? Both these have chemosensitive areas or receptors to CO2 and H positive concentration. Now, signals coming from these receptors go to the rhythm center, and that is how your rhythm is generated, whether your breathing rate has to be increased or decreased. So more CO2, more H positive ions, more breathing rate. Less CO2, less H positive ions, low breathing rate. So very important, oxygen plays very low role. So role of oxygen in regulation is quite insignificant. It is basically your CO2 and H positive ions which are going to determine your breathing rate. Are you understanding, everyone? Now coming on to the disorders and the last part of the human physiology, asthma. is the difficulty in breathing, characteristic feature wheezing sound, and there is inflammation of bronchi and bronchioles. Emphysema, chronic disorder in which alveolar walls damage, so respiratory surface decreases, occurs mainly because of cigarette smoking. Then occupational respiratory disorders, they are because of some specific occupations which involve some very minute fibers which can settle down in your lungs, so like grinding and stone baking or dust, right? So body is not able to cope up, and long exposure gives rise to inflammation. There is fibrous development known as fibrosis, and it is a serious lung damage. So according to the Factory Act, workers in the industries should wear protective masks. So basically the owners have to provide them. So guys, with this we complete human physiology. Let's have a good clap. Yes. Yes, I'll have a sip of water. Give me a thumbs up that you're all done with human physiology. Let's all clap for ourselves. 77% of CO2 is transported through plasma. Yes, Diganta, you're very, very right. 70% through bicarbonate ions and 7% through plasma. Very good, very good. Diganta, your NEET 2026 is definitely clear. Campbell, I will explain Bohr and Haldane effect in my unit-wise revision. Okay, don't worry about that. I'll explain that. I'll explain that properly. Don't worry. So make sure that you write down this. You make a list of this. Thank you, Jyoti. Okay, guys, moving on to now plant physiology. Plant physiology. Coming on to plant physiology. So first chapter is plant growth and development. Now see, plant growth and development. See, what is growth? Irreversible permanent increase in the size of the organ or even the individual cell. And growth is an active process. It occurs at the expense of energy. Now plant growth is special that it is indeterminate. What is indeterminate? Means the growth is not limited, it is unlimited. Plants can keep growing throughout their life. Why? Because they have presence of meristems at certain locations. Assertion reason, yes or no? This is assertion reason. Now see, dicot plants, gymnosperms, the lateral meristems, vascular cambium, cork cambium, these are all the meristems which appear later in life. Is that clear? So these cambiums, these lateral meristems help in increase in the girth, and so we call this growth as secondary growth. Now growth can be measured. Okay, so at cellular level, growth means increase in the level of amount of protoplasm. Now these are very important lines. In case of maize seed, in one single maize, root apical meristem can give rise to more than 17,500 new cells per hour. This was a PYQ. This was a PYQ in the match the following. Please note. So in maize, we measure growth in terms of increase in the number of cells. So in every hour, 17,500 cells increase in case of one single maize. And in case of watermelon, the cell size increases. So growth can be measured on so there are various parameters in which growth can be measured. Increase in the size of the cell, increase in the number of the cell, volume of the cell. So in watermelon, the size can increase up to 350,000 times. Just imagine. Okay, for pollen tube, it is the length of the pollen tube and so on. Now there are three phases of growth. So for meristematic area, see, what are the characteristics of meristematic cells? So the cells in the meristem, they are always rich in protoplasm. They have conspicuous, means well developed nucleus. The cell walls are thin and primary and abundant plasmodesmatal connections. These are the cell junctions in plant cells. Is that clear? Now, second is the phase of elongation. After meristem, the cells goes in the second stage, which is the second zone, the phase of elongation. Now, in phase of elongation, the cells now undergo vacuolation. So, large vacuoles are formed which will cause cell enlargement and new cell wall depositions will take place. Then, beyond phase of elongation is the phase of maturation. What will happen during phase of maturation? Cell wall thickenings will take place and protoplasmic modifications will occur. Please let me know that these points are clear to all. Please let me know that these points are clear to all. Clear? Now coming on to the growth rate. There are two types of growth rates that are observed, arithmetic and geometric. See, what is arithmetic? There is sequential increase, 1 to 2, 2 becomes 3, 3 to 4, 4 to 5, and so on. This is arithmetic. And what happens in geometric? So there is sudden jump. Like 1 becomes 2, 2 will give rise to 4, 4 becomes 8, 8 becomes 16, and so on. This is geometric. Now, one very important line. What about zygote? What kind of division, what kind of growth rate is shown by zygote? So it is clearly written here, stages during the embryonic development. So embryonic development shows both geometric and arithmetic phases. See, so embryonic development shows both geometric and arithmetic type of phases. Now see, these are the two graphs. These are the two graphs. So for arithmetic growth, see, in arithmetic, I told you there will be gradual increase, 1, 2, 3, 4, and so on. So what happens in mitosis? As we know, one mitotic cell will give rise to two cells. Now, mitotic cell division, one of the daughter cells will divide, but the other will differentiate. So only one continues. Out of the two, only one continues to divide, right? So this is arithmetic growth rate. So that is why, since there is gradual increase, we get a linear curve. And what is the equation? LT is equal to L naught plus RT, right? While in geometrical growth phase, since there is sudden jump, 1, 2, 2, 4, 4, 8, 8 becomes 16, then 32, and so on. So initial growth is slow. It is known as lag phase. See the graph. Initial phase is slow phase or lag phase, but then it is followed by sudden change, sudden increase, known as exponential phase or the log phase. And then when the nutrient supply, since the nutrient supply is limited, and towards the depletion of the nutrients, we get a stationary phase. Is this clear? We get a stationary phase. And what is the equation? W1 or WT is equal to W naught ERT. So here we get this logarithm. So what is E? E is the base of natural logarithm. What is R? This was a PYQ. What does R indicate? R indicates the relative growth rate and is also the measure of ability of the plant to produce the new material. Please let me know that you are able to understand all these things quickly. Yes, please. Please let me know that these points are clear to all. Okay, moving on to, so R is also the relative growth rate. It is also known as the efficiency index. It is also known as the efficiency index. Is that clear? All right. Now coming on to these two, absolute growth rate and relative growth rate. Absolute and relative growth rate. Now see, very important. Just take the example. See, if we go by the definition, so measurement and comparison of the total growth per unit time is the absolute growth, and the growth of the given system per unit time expressed on common basis, when there is a common basis using the initial parameter, that is the relative growth rate. Now, for example, in this given picture, see, in this first diagram, the smaller leaf smaller leaf has surface area of 5 centimeters square. Then it grows up to 10. So what is the absolute growth? That is 5 centimeters square. But what is the relative growth? So 10 minus 5 upon 5. So 5 by 5 is 1. Is that clear? So relative growth will be equal to 1. But now if I talk about this larger leaf, so initial is 50, final is 55. So absolute growth rate would be 55 minus 55. But the relative would be 5 upon 50. That will be 1.10, 1 upon 10.1. So what is the formula for absolute growth rate? Absolute growth rate is final minus initial. Simple. Please write down. Final minus initial is the absolute. But final minus initial upon initial becomes the relative growth rate. Are these points clear? Are these points clear? Now, what are the conditions required for growth? Water, oxygen, nutrients, essential. Water helps in the turgidity of cells, leading to the extension or the growth. Also, water is important medium for enzymatic activities. Oxygen is needed for producing energy, for metabolic energy. Nutrients can be needed in higher amount, known as macro elements. Those which are needed in the lesser amount are called micro elements, and they are important for the synthesis of protoplasm. Then temperature should also be optimum. Environmental signals like light and gravity will also affect the stages. Then we have 3 different stages. We have already discussed yesterday. Differentiation, in which major structural changes both in cell wall and protoplasm will take place. right? Then we have dedifferentiation, in which the permanent cells will regain the capacity to divide and become meristematic again. We have two examples here, interfascicular cambium and cork cambium. And then after these, after dedifferentiation, when the secondary meristem, see, after dedifferentiation, the meristem which becomes is known as secondary meristem, it will again become mature, and this process is known as redifferentiation. So the tissue, permanent tissue which is formed after redifferentiation is known as secondary tissue. Have or have we not discussed it yesterday? Please let me know. How many of you have attended my previous unit-wise revision of Unit 2? How many of you have attended that? Because those of you who attended that, I have discussed this topic already in detail. Please let me know. Yes, please. You have? Nahi? Buri baat. Gadbad. Then coming on to development. Development is a term that includes all changes that an organism goes through in the life from germination to senescence. Now this sequence, this was in PYQ. So meristematic cell undergoes plasmatic growth, differentiation, expansion and maturation. The four things happen to convert the meristematic cell into the mature cell, right? After maturation, it undergoes senescence, that means aging, leading to death. Clear? Okay, now plants follow different pathways in response to environment. Now see, this ability to behave differently in different conditions is known as plasticity. For example, heterophyly. So when the leaves show these changes, this is known as heterophyly. So heterophyly is an example of plasticity. So what is heterophyly? Two different types of leaves will be present in the same plant. Okay, now this heterophyly can be based on the stages of life, or it can be due to the environmental difference. For example, in cotton, coriander and larkspur, heterophyly is stage based. So leaves of juvenile, see here in this figure, this is the picture of larkspur. So in juvenile stage, the picture of the, look at the picture of the leaf, and then in adult stage, look at the picture of the leaf. Are you able to see? So different stages have different types of leaves. This is heterophyly due to different stages. But then heterophilous development can also occur due to environmental change. For example, if the leaf, see, this is an example of buttercup. So leaf shape in the terrestrial habitat is like this, but when this buttercup grows in the aquatic medium, the shape of the leaf becomes highly dissected. Is this clear? So this is heterophyly. So you must know the examples. Larkspur, cotton, coriander for show heterophyly based on their stage of life, and buttercup shows heterophyly based on the environment. So overall development is sum of growth and differentiation. There are intrinsic and extrinsic factors which affect that. What are the intrinsic factors? Intrinsic are the growth regulators and the genetic factors, and extrinsic factors are the light, temperature, water, oxygen and nutrition. Now coming on to the plant growth regulators, very important. So this paragraph which I have highlighted, this shows the uh, um, derivation. So this shows that what is the basic chemical composition of your plant growth regulators. So indole compounds are your auxins, adenine derivatives are your kinetins or cytokinins, right? Then your carotenoid, your abscisic acid is derived from carotenoids. Then gibberellic acid is obtained from terpenes. This is your PYQ, and ethylene is a gaseous compound. Is that clear? Then there are plant growth promoters. Three examples of growth promoters are auxins, gibberellins, and your cytokinins. While your abscisic acid and ethylene, these come under the category of plant growth inhibitors. Are you clear with this? Now coming on to the discovery. See, talking about auxin. Auxin was discovered by Charles Darwin and his son Francis Darwin. Please understand, there is confusion sometimes. Students are generally confused. What is the role of the invent? So Darwin and his son, they discovered it in the coluptile of canary grass, and they performed this experiment, unilateral illumination of this coluptile. So they observed that the tip of the shoot grows towards the light. This is photoperiodism, positive photoperiodism, and this positive photoperiodism is due to auxin, but they were not able to isolate it. So auxin was first isolated by F.W. Wendt from the tips of oat seedling. Oats the same oats that we eat in a breakfast, right? Oat is also known as avena. Now, second is the discovery of gibberellic acid by E. Kurosawa. From this fungus, Gibberella fujikuroi, this fungus caused bakane or foolish seedling disease in the rice. Clear? Then Skoog and his co-workers, they discovered cytokinins from the extracts of vascular tissue, yeast extract, and coconut milk. But it was crystallized by Miller. So Miller was able to crystallize this substance in the form of kinetin. Clear? Then three independent researchers discovered ABA. They all named them differently. One called it inhibitor B, abscission 2, and the third one called it dormin. But later on, when it was chemically proved to be the same thing, we named it as abscisic acid, ABA. And then H Cousins, they discovered this volatile substance released from ripened oranges known as ethylene. And it was seen that ripened oranges are releasing some gaseous substance which is able to ripen the unripe bananas. Are these points clear to all? Points clear to all? Then coming on to the effects. Now see, important lines I have underlined here. Auxins. The natural auxins are IAA, indole acetic acid, and indole butyric acid. And then NAA, naphthalene, acetic acid, and 2,4-D. These are the synthetic auxins. Auxins promote flowering in pineapples. Growing apical buds inhibit the lateral buds. This is known as apical dominance. And removal of shoots, tips, that is decapacitation. It results in the growth of lateral buds, and this is widely used in tea plantation and hedge making. Then auxins induce parthenocarpy. Auxins induce parthenocarpy. This has just jumped onto the. Okay. Yes, auxins induce parthenocarpy in tomatoes. 2,4-D is used to kill the dicot weeds only, not the monocot weeds at all. It will only kill the 2,4-D will only kill the dicot weeds. Is this clear? Then talking about gibberellins. Gibberellins, they're acidic, more than hundreds are known. Then increase the length. So whenever there is increase in the length of the stalk or of the grape stalk or the length of apple fruit, that is all because of gibberellin. Increase in the length of grape stalk, elongate the shape of fruits like apple, they delay senescence, they increase the malting process in brewing industry, alcohol production. So this is all gibberellin. Now, if gibberellin is sprayed on sugarcane crop, it can increase the length of the stem so much that the yield can be increased as much as 20 tons per acre. This is the effect of gibberellin. Then spraying gibberellins on juvenile conifers will hasten the maturity period and early seeds will be produced. Then it will also promote the bolting process. What is bolting? Internodal elongation prior to flowering. And where bolting is seen in beets, cabbages, and many rosette plants. Clear? Cytokinins were discovered as kinetin, but from an autoclaved herring sperm. Herring is a fish. So first time kinetin was discovered from the sperm of a fish known as herring. Kinetin doesn't naturally exist in plants. In plants we get cytokinin in the form of zeatin. So zeatin is the form of cytokinin in plants. We obtain it from corn kernels and coconut milk. Is it clear? So synthesized actively and maximally in the place where rapid cell division is occurring, that is in the meristematic areas. So cytokinins are antagonistic to auxins because auxin causes apical dominance. Cytokinins helps in overcoming the apical dominance. Are these points clear? They promote the nutrient mobilization, delay the leaf senescence. Then there is horizontal growth of seedling, and because of ethylene, apical hook formation. Respiration rate increases, and this is known as respiratory climactic. This is all because of ethylene. It breaks the seed and bud dormancy. Germination in peanut seeds and sprouting of potato tubers is caused. Then it causes fruit set in pineapple. Flowering is because of auxin. Fruit set is because of ethylene. Induces flowering in mango. Now, form in which ethylene is used is called ethaphon. This is a tablet dissolved in water, and then aqueous solution is sprayed to hasten the fruits ripening in tomatoes and apples. It causes thinning of cotton, cherry, and walnut, and female flowers in cucumbers. Is it clear? ABS stimulates the closure of stomata and increases the tolerance towards the stress, and that is why it is known as a stress hormone. It is antagonistic to GA. Cytokinin and auxins are antagonistic. ABA and GA are antagonistic. Now, role of PGA is one kind of intrinsic role. The second intrinsic control is because of genome, the genetic factors. Okay, and extrinsic factors I've already discussed: temperature, light, and water, and oxygen, and nutrients. Are these points clear? So, first chapter of human physiology is done. Quickly give me a thumbs up that we are done with all this. Now, talking about respiration in plants. So we already know what is cellular respiration. That is when actually the food will be broken down to release energy. And this energy is trapped in the form of ATP. Now, this is a PYQ line. It is very, very important. See, this line has come in, I think I'll tell you exactly. This line was in which year? Yes, this line, the breaking of CC bonds of complex through oxidation within cells leading to the release of considerable amount of energy is respiration. This was 2024. This was line was from 2024. And the products which are broken, sorry, compounds which are broken down are known as respiratory substrates, which are usually carbohydrates, but proteins, fats, and organic acids. These are all used to release energy. Are these clear? Now, energy is not released in just single step because otherwise it will be difficult for us to contain it. So it is released in slow stepwise reactions with controlled. These reactions are controlled by enzymes and energy is trapped in the form of ATP. Are these points clear to all? Then moving on, that see, most of the living cells are located close to the surface in plants. Okay, like in case of leaves. So living cells are in thin layers, and so that oxygen can reach to the thin layers, there are certain openings known as lenticels. Is that clear? Most of the interior part of the plants are dead and only for the mechanical support. Coming on to glycolysis, it was studied by this whole process. The scheme was studied by Emden, Meyerhoff, and Paranas. That is why it is known as EMP. Now, in anaerobic organism, this is the only process and occurs in the cytoplasm. This process is common to all the living organisms. Here, partial oxidation of glucose takes place and one glucose will form how many molecules of pyruvic acid? 2. In plants, the source of glucose is sucrose. So first of all, plants will break down sucrose into glucose and fructose using the enzyme invertase or sucrase, right? Now, this line was again in your PYQ. So all these lines that I have marked here are PYQs. Guys, please understand. These lines are all PYQs. Is it clear? Glucose and fructose. Glucose and fructose are phosphorylated to give rise to glucose 6-phosphate by the activity of the enzyme hexokinase. This was 2019 ka question. Now, overall glycolysis, how many steps are there? There are 10 reactions. Is this clear? Then this line was in 2024 that DPGA to 3PGA is an energy yielding process. So in this whole reaction, you have to know that which step is going to use ATP, which all steps. So there are 2 steps. Glucose to glucose 6-phosphate will use ATP. Fructose 6 to fructose 1,6 will again use ATP. But thereafter, there will be energy yielding process. So what are the steps which yield energy? You should be knowing that clearly. 2-PEG, 2-PPA, this phosphoenol pyruvate, PEP. 2-PEP to pyruvic acid is energy yielding, and similarly 1,3-bis-phosphoglyceric acid to 3-phosphoglyceric acid. This is also energy yielding. So you must know that. Now, then there is this question again. This question was asked in 2022. Can you then calculate how many ATP molecules are directly synthesized? See, one glucose molecule will use 2 molecules of ATP and give 4 molecules of ATP. Use 2 and give 4, produce 4. So net gain of 2 ATP molecules occurs in one glycolysis cycle. Is it clear? So answer here is 2 ATP. So one glucose will produce how many pyruvic acid? 2 pyruvic acid. So go through this whole process clearly. I will be discussing it in detail in my unit-wise revision also. Is this clear? Then moving on to the next step. So once this pyruvic acid is formed, this pyruvic acid can have different fates. It can enter 3 different pathways. It can enter the lactic acid fermentation or alcoholic fermentation, or it can go into the aerobic respiration. In case of anaerobic conditions, it will go for lactic acid or alcoholic fermentation. Only in the presence of oxygen, complete oxidation will take place, of complete oxidation of glucose will take place to form CO2 and water, and this requires Krebs cycle. And this is the aerobic respiration. Now, talking about fermentation, where does it take place? See, by yeast in anaerobic conditions and leads to the formation of carbon dioxide and ethanol, resulting in the production of pyruvic acid. It requires two enzymes. See here, alcoholic fermentation will require two enzymes: pyruvic acid decarboxylase and alcohol dehydrogenase. Clear? While there are some bacteria and our muscles which can respire anaerobically too. And in that case, bacteria and our muscles, they are going to produce lactic acid from pyruvic acid. Is that clear? And the enzyme used here is lactate dehydrogenase. Lactate dehydrogenase. Now, very important line, not much energy is released and less than 7% of energy in glucose is released during this reaction. Now, this was 2022. These lines were in 2022. Is this clear? Then, yeast poisons themselves beyond 13%. So once alcohol is being produced, maximum concentration which can be produced out of this from this reaction is 13%, right? Why? Because beyond 13% of alcohol, the acidic conditions increase so much that the yeast is not able to survive beyond that. Okay, so the maximum concentration of alcohol obtained by natural fermentation is only 13%. If you want to go beyond that, we have process called distillation. Is this clear? Then these lines are super important. They were in 2016, they were in 2018, they came in 2024 and 23. So NTL loves these lines. What are these lines? That aerobic respiration, complete oxidation of pyruvate or pyruvic acid by stepwise removal of H positive ions, leaving 3 molecules of carbon dioxide. This is known as Krebs cycle, and this occurs in the matrix of mitochondria. While the second step written here, the passing on of electrons moving from part of hydrogen atoms to molecular oxygen with simultaneous synthesis of ATP, this occurs in the inner membrane of mitochondria. What are we talking about? We are talking about ETS. So first step is Krebs cycle, second step is your ETS. The Krebs cycle takes place in the matrix and ETS occurs in the inner membrane of mitochondria. Are these points clear to all? Then this whole paragraph was in 2023. Which are the enzymes used? So this is the link reaction. See, before entering into the mitochondria, really, pyruvic acid has to undergo some change. This is known as oxidative decarboxylation. So pyruvic acid is a 3-carbon compound. It will release carbon dioxide and become acetyl coenzyme A, plus CO2 is released, NADH and H positive. So NADH is also produced here. See, so this is the link reaction which connects glycolysis to Krebs cycle, and 2 molecules of NADH are produced here from 2 molecules of pyruvic acid, because one glucose gives you 2 pyruvic acid. All right. So 2 pyruvic acid will give you 2 NADH. So in this reaction, we are showing for 1 pyruvic acid. So in reality, we have 2. So as a result of this link reaction, at the end of the link reaction, we'll get 2 molecules of NADH. Is this point clear? Then coming on to Krebs cycle, which is also known as tricarboxylic acid cycle, also known as citric acid cycle, because the first compound formed is a citric acid. Is this clear? Now this whole cycle is very important. Whole cycle is very important. All these lines are PYQs, guys. All these lines. So during the conversion of succinyl coenzyme A to succinic acid, GTP is produced. So now in this reaction, you must know that at which step NADH are produced, which step involves the production of GTP, and which step involves the production of FADH. So clearly written and seen, NADH is being produced at 3 steps, FADH is produced once, and GTP is produced once. So this is for 1 pyruvate to 1 acetyl coenzyme A, and it enters 1 Krebs cycle. But in actuality, we have 2 pyruvic acids coming from 1 glucose. So how many Krebs cycle will be running from one? Will be running? That is 2 Krebs cycle. Is this clear? So we'll be able to calculate the energy here. We'll be able to calculate the energies here. All these are PYQs, guys. We got so many questions in 2022, 23, 24, 20, 18. So hardly a year which has gone by without getting a question from this topic. Then finally, when this enters the, so this is a final reaction of the Krebs cycle. This is a final reaction occurring in the mitochondrial matrix. Are these points clear to all? So coming on to the final step of aerobic respiration, which is the electron transport system, which occurs in the inner mitochondrial membrane. Now you have to remember, first thing first, all the complexes. There are 5 complexes used. The name of the complexes are important, and we get direct questions out of it. So learn the name of the complexes first. Then you must know where are these complexes present. These complexes are present in the inner mitochondrial membrane. Okay, except for the complex number 2, which is the succinate dehydrogenase. This is towards the matrix. Now, complex number 2 is common between Krebs cycle and ETC. Now, these lines are all NCERT. These are all previous equations. Where is ubiquinone present in the inner membrane? Whereas cytochrome C, it is a small protein attached to the outer side of inner membrane and acts as a mobile carrier. Now, complex 4 has 2 copper centers. Then ATP synthetase is a complex 5 and helps in the production of ATP. Now, one NADH is equivalent to 3 ATP molecules. One FADH2 is equivalent to 2 ATP molecules. Now, we call it aerobic because it uses oxygen, but role of oxygen is limited to the terminal stage. So, oxygen is like that showstopper which comes at the end, but the whole function or the whole program is in the name of the showstopper. So, similarly, the oxygen. So, phosphorylation, photophosphorylation. Now, listen to this line. This is PYQ 2024 and 2021. This blue line. Unlike photophosphorylation, in which light energy is utilized for the production of proton gradient, in respiration, we don't use light energy. In respiration, the energy of oxidation and reduction is used. That is why we call it oxidative phosphorylation. See, production of ATP is phosphorylation. If you use light energy to produce ATP, it is photophosphorylation. If you use the energies of oxidation and reduction reactions, we call it oxidative phosphorylation. Is that clear? So this was ETC. Now very important, this F naught. See, this is the complex number 5, which is ATP synthetase. It has 2 components, F naught. F naught is an integral membrane protein, acts as a channel protein through which protons can pass, and 4 protons will pass through F naught, and passage of 4 protons will produce 1 ATP. Is that clear? All right. So guys, this was a balance sheet, and then amphibolic pathway. Why? Because basically in respiration, both anabolism and catabolism take place, right? For example, fatty acids can be broken down to form acetyl coenzyme A, as well as in some other situation, acetyl coenzyme A can also form fatty acids. So since both ways, the reactions can occur in both ways, simple to complex and complex to simple. That is where we prefer to call respiratory pathway as a amphibolic pathway. And then this interrelationship, very important. So carbohydrates we know form pyruvic acid, and pyruvic acid forms acetyl coenzyme A. Proteins will break down into amino acids, and amino acids will enter the pathway by forming pyruvic acid, right? And then fats will break down into fatty acids and glycerol. Glycerol will form DHAP, which will enter the glycolytic pathway, but fatty acids will change into acetyl coenzyme A. So you can get multiple questions. Now out of these three, within these three, fats, carbohydrates, and proteins, one common byproduct is the acetyl coenzyme A. Is that clear? But for carbohydrates and proteins, even pyruvic acid is a common byproduct. Is that clear? For glycerol also. But if we consider all the three, then it is acetyl coenzyme A. Is this point clear to everyone? Please let me know. Then the values of respiratory quotient. For carbohydrate, it's 1. For fats, it's less than 1. For tripalmitic acid, see the formula, it is 0.7, and for proteins, it is 0.9. While for organic acids, it's more than 1. It's an important line. Pure proteins and fats are never used as respiratory substrates. Please let me know. Thus, this chapter is also clearly read by you with me. Quickly let me know. Guys, I really appreciate, but I also understand that this can only happen, when we are working together, when we are reading together. So we are on the last chapter of plant physiology, coming on to plant physiology. Last chapter, that is photosynthesis. See, Let's talk about some discoveries here. So when we experiment with the variegated leaf is the one which is multicolored. When variegated leaf is exposed to light, we through this experiment, we have done this experiment in lower classes, in class 10 and all. So through this experiment, what do we prove? That photosynthesis occurs only in the green parts of the leaves. So variegated leaf experiment proves that chlorophyll is necessary for photosynthesis and it occurs only in the green parts. Then there was another experiment when leaf was enclosed in the test tube containing KOH, and we know that potassium hydroxide absorbs CO2. So this experiment will prove that CO2 is required for photosynthesis. Then there are some other experiments. Joseph Priestley, he discovered oxygen. He worked through this bell jar experiment. And what did he conclude? He concluded that plants restore the air, whatever breathing animals and burning candles remove. Clear? Then, using the same setup, but Jan Ingenhouse, he discovered that sunlight is essential for plants and he gave the term photosynthesis also. Ingenhouse performed this experiment with aquatic plant in which he observed that small bubbles were formed in the green plants only in the bright sunlight but not in the dark. So he said that sunlight is important. Then Julius von Sachs, he gave evidence that production of glucose occurs, and glucose is formed during photosynthesis. Second, this glucose is stored as starch, and later on he also showed that green substance, that is chlorophyll, is located in special structure called chloroplast. Three things: one, glucose is produced; second, it is stored in the form of starch; and third, that chlorophyll is stored or present in chloroplasts. Then Engelmann, he did this prism experiment. He splitted this white light and then illuminated the green alga. Which alga did he use? Cladophora. So in different regions of light, Vibgyor, he saw that different amount of bacterial growth was seen. So Cladophora. So bacteria was used to detect the site of oxygen evolution. So first action spectrum basically was built by Engelmann. Is that point clear? Was built by Engelmann. Then we have Cornelius van Niel. So Van Niel experimented on purple and green bacteria, and he showed that photosynthesis is an light dependent reaction, number one. And second, the source of hydrogen is going to determine whether oxygen will be produced or not. And hydrogen from a suitable oxidizable compound will reduce the CO2. Is it clear? Green plants use water as a hydrogen donor and that therefore produce oxygen, while purple and sulfur bacteria use H2S as a hydrogen donor and therefore they don't produce oxygen, instead they produce sulfur. Is that clear? Therefore he concluded that only when water is used as a hydrogen donor, oxygen is produced. So oxygen evolves from water and not from CO2. So out of CO2 and water, the source of oxygen is water. Is this point clear? Then coming on to the light and dark reactions. See, light reactions, photochemical reactions; dark reactions, carbon reactions. In stroma, there are enzymatic reactions. So dark reactions occurs in stroma, while light reaction occurs in thylakoids. Now very important. If we perform a paper chromatography with all these pigments, see these colors are important. We have got these colors in match the following multiple times. So chlorophyll A has to be matched with bright and blue color. Chlorophyll B shows the yellow green color in chromatograph. Then xanthophylls will have yellow color and carotenoids will have yellow to yellow orange. This match the following is very, very important. Now out of all the pigments, which is the most abundant pigment? It is chlorophyll A. Is that clear? Now, which color shows maximum photosynthesis? Answer is red and blue. See, maximum absorption of chlorophyll is in blue and red light. But if you have to choose between red and blue also, the answer is red. Is that clear? And minimum photosynthesis occurs in the green light. Are these points clear? All these are PYQs. Light reactions, also known as photochemical reactions. What all happens during light reaction? Number one, light is absorbed. Second, water splitting will take place. Then, if water will split, oxygen will be produced. And fourth, high energy chemical intermediates are produced. Four things that happen during light reaction. What are the high energy intermediates? ATP and NADPH. Is this clear? So for absorbing light, we have this pigments are organized in this light harvesting complex. Okay. So in photosystems, we have these light harvesting complex. These light harvesting complex are also known as antenna, and they are made up of pigment molecules and proteins. So remember, light harvesting complex is only known as antenna, and it has pigment molecules as well as proteins. Okay. And then photosystem. See, photosystem has two things, light harvesting center and reaction center. Okay. So light harvesting center is formed of pigment molecules and proteins. Now these pigments are other than chlorophyll A. Chlorophyll A will form the reaction center. And in reaction center, there is single molecule of chlorophyll A. Now, based on the type of chlorophyll A, we have two types of reaction centers. Now, one chlorophyll A can absorb to the maximum of 700 nanometer. Is it clear? So the chlorophyll A, which can absorb maximum up to 700 nanometer, such photosystem is known as photosystem 1. It was first discovered. But the photosystem 2, which was discovered later, has chlorophyll A with maximum absorption at 680 nanometer. Is this point clear? Now, coming on to the electron transport. Very important. So we have two types of transports, electron transport. One is the Z scheme, another is the cyclic transport. So in Z scheme, see this diagram. In Z scheme of light reaction, basically, so why do we call it Z? So basically it forms the shape of Z. So in Z scheme, first the electron moves upwards, then downwards, then upwards. This was a PYQ. What is the correct direction of electron flow? Upward, downward, and then upward. Is this clear? Then in this Z scheme, first we use PS2 and then followed by PS1. Is that clear? So PS2, the use of PS2 is followed by PS1. Are these points clear to everyone? Is this clear? Now, Z scheme, it is because of the characteristic shape. Now, in Z scheme, as I said, we use PS2 first. So, how is PS2 continuously being supplied due to the splitting of water? So, water splitting is associated with PS2 and not with PS1. Where is this water splitting complex present? On the inner side of membrane of thylakoid. Are these points clear? Now, during these electron flows, see, energy will be produced. Energy will be produced. Now see, during this Z scheme, the electron which has gone upwards and then downwards and finally upwards again, it is not coming back. So the energy produced or ATP produced during this Z scheme will be known as non-cyclic photophosphorylation. Is that clear? So non-photocyclic phosphorylation is equivalent to Z scheme. So ATP production during Z scheme is non-cyclic. Is that clear? But then we have cyclic. And in Z scheme, we are using both the photosystem. First PS2, then PS1. Now, in this figure 11.6, we see the cyclic photophosphorylation. In cyclic, we only use PS1. So electron which goes up comes back. So we don't need any external source of electron here. See, when two photosystems work in series, first PS2 and PS1, that is non-cyclic in Z scheme. But when only PS1 is functional, it is circulated back within the photosystem. So cyclic flow of electrons is occurring. So energy production, ATP production is also known as cyclic phosphorylation. Now this occurs in the stromal lamella. Is that clear? Stromal lamella. Sorry. Where this is happening? While yes. So stromal lamella or the Fred membranes has only cyclic photophosphorylation because they only have PS1. While membrane or lamella of the granule or granule lamella will have both. So granule lamella will show Z scheme and stromal lamella will show cyclic. Is that clear to all? Please let me know that this point is clear to everyone. Now, regarding the ATP production. So basically ATP production is going to occur because of chemiosmotic hypothesis. It is due to the development of proton gradient across the membrane. Is that clear? Is that clear? Now, splitting of water will occur on the inner side of the membrane. Hydrogen ions will be produced as a result of the splitting. These ions are going to be these ions are going to be accumulating here in the lumen. See, this pink membrane represents the thylakoid membrane. Clear? So water splitting occurs here. The oxygen is produced, diffused out, H positive ions get concentrated in the lumen. So primary acceptor of lumen, these lines are all NCERT, please understand. The primary acceptor of electrons with located on the outer side of membrane, not to the electron carrier but to the H carrier. Now, NADP reductase enzyme, where is it present? On the stromal side. Very important. Now, electrons come from the acceptor of electrons of PS1. Protons are necessary for this. Protons are removed from the stroma. Protons in the stroma decrease in number and lumen increase. While in the lumen, it increases. So, since a gradient is set, the difference is set, right? So, this difference will help in ATP production. Now, what are we going to do? We are going to break this gradient. Very important. See, ATP production is not because of proton gradient. ATP production occurs because of breakdown of the gradient. Is that clear? So, due to the movement of protons through, again, complex number 5, just because now it is present in chloroplast, we'll call it CF0. Okay? CF0 and CF1 of ATP synthase. Is this clear? So CF0 is always the thylakoid, always embedded in the membrane, in the thylakoid membrane here. So transmembrane channel allows facilitated diffusion both here as well as in mitochondria, while CF1 is the one which protrudes towards the outer side, outer surface of thylakoid membrane. Very, very important. Please see here. What does chemiosmosis require? This is a PYQ. For chemiosmosis, we require one membrane, which is a thylakoid membrane, a proton pump, okay, then a proton gradient, a difference, and ATP synthase. These are the 4 things needed for chemiosmosis to take place. Are these points clear to everyone? Please let me know. Okay, now ATP will be immediately used in the biosynthetic reaction. Please let me know. This biosynthetic reaction is another term for ATP. Now, how did we find the pathway of a dark reaction? It was by use of radioactive C14. Okay, and who did that? Calvin did that. So we call it Calvin cycle. Clear? The first product of Calvin cycle is 3-PGA, 3-phosphoglyceric acid. Now, since this is a 3-carbon compound, we call this cycle as C3 pathway also. Is this clear? So, C3, C2, C4, all are based on the first stable compound formed. Is that clear? In C3, 3-carbon compound, that is PGA, will be formed. In C4, 4-carbon compound, oxaloacetic acid will be formed. These are clear-cut questions coming in match the following. So you should make a list of all these compounds. Which compound is 2-carbon, 3-carbon, 4-carbon? Is this clear? Alright, PGA was the first compound of carbon fixation. Now, but if we have 4-carbon compound as the first stable compound, which is oxaloacetic compound, we call that a C4 pathway. Is that clear? Now, in C3 cycle, which is the carbon dioxide acceptor? It is a 5-carbon sugar, which is ribulose bisphosphate, RUBB. But in C4 cycle, it is a 3-carbon compound, which is PEP. Is that clear? So C3 cycle occurs in 3 steps. So this is a PYQ. What are the 3 steps? In order: carboxylation, reduction, and regeneration. And see, it is in the alphabetical order also. So carboxylation is CO2 utilized in carboxylation. What happens? CO2 is being utilized. CO2 is utilized, and who takes this CO2? It is RUBP. This reaction occurs in the presence of RUBP carboxylase. We like to call it Rubisco because it is not only carboxylase, it is oxygenase also. It shows oxygenase activity also. Coming on to this figure, very, very important. We got this flow chart directly in a PYQ where you had to fill in these blanks, which is the first step, second step, and third step. If you have solved PYQs enough times, you know this. How many of you have seen this question in the PYQ? Quickly let me know. So first step is RUBP taking carbon dioxide. So this is carboxylation and becoming 3-PGA. Then 3-PGA will get reduced to form triose phosphate. Now this step is known as reduction, and very important, each reduction step will use 2 ATP and 2 NADPH. Very, very important. Very, very important. And then this triose phosphate will regenerate to form RUBP, and the step will require 1 ATP. So in all, we use 3 ATP and 2 NADPH. Is that clear? So 2 ATP during reduction, 1 ATP during regeneration, and 2 NADPH during reduction, and nothing during any other reaction. So these lines are very, very important. This is very, very important. So see, if we have to fix, so in order to produce one glucose molecule, we have to fix six molecules of glucose. So when we have to fix six molecules of CO2. So six molecules of CO2 will require 3 into 6, 18 ATP, 2 NADPH, 2 into 6, 12 NADPH. Are these points clear? These lines which I have underlined are all PYQs. See, then C4, I've told you, first compound will be oxaloacetic acid. Now C4 plants will show special anatomy known as Krenz anatomy. Basically here, vascular bundles are surrounded by bundle sheath cells. We have already discussed it yesterday also. Can you name any C4 plants? Mostly monocots, but some dicots also. Two examples are maize and sorghum. But remember, rice is not a C4 plant. Wheat is a C4 plant. Is that clear? So here, coming directly, so in C4 cycle, the primary CO2 acceptor is PEP, which is a 3-carbon compound. So 3 and 1 carbon dioxide, it will result in the formation of 4-carbon compound, which is oxaloacetic acid. Now all this is going to occur in the mesophyll cells. See, basic difference between C4 and C3 for with respect to the position of rubisco. In C3 plants, rubisco will be present in mesophyll cells. But in case of C4 plants, rubisco is not in the mesophyll cell. Rubisco will be found in the bundle sheath cell. Now, this figure is very, very important. The upper cell is a mesophyll cell. See, upper cell is a mesophyll cell. Where PEP is present, PEPcase will be the enzyme used for the fixation. First C4 acid or OAA will be formed. Is that clear? Changing into malic acid, getting transported into the bundle sheath cell. So rubisco is found here in the bundle sheath cell. Is that clear? So here the number of ATP molecules used increases tremendously. In C3 cycle we use 18 ATP, while in C4 we are going to use 30 ATP. Is that clear? Then we have the C2 cycle, which is an entire wastage, photorespiration, in which rubisco is going to add oxygen, showing a wasteful process. Oh, is that clear? This is photorespiration. It is a wasteful process because there is nothing useful produced. Nothing useful is produced. We call it C2 cycle because RUBP is converted into two molecules of PGA and instead of forming two molecules of PGA, it forms one molecule of phosphoglycerate, PGA, and one of phosphoglycolate. Is that clear? So since there is a 2-carbon compound involved here, which is phosphoglycocolate, we call it a C2 cycle. Are these points clear? Then C2 all. Is this point clear to all? Please let me know. So they show better tolerance. Are these points clear to everyone? Now coming on to the factors. The external factors involved are sunlight, temperature, CO2 and water. This is law of limiting factors given by Blackman, where he says that if a chemical process is affected by more than one factor, then the factor which is in the least amount is going to determine the speed. Is that clear? Then this is the graph showing the graph of light intensity and the rate of photosynthesis. So linear relationship is seen between light and CO2 at the low light intensity, and that at higher intensity we class. At higher light intensity, the rate does not show further increase. Is that clear? Then there is 10% law which says that light saturation occurs at 10% of the full light. Light saturation occurs at 10% of full light. Is that clear to all? Hanji, are these points clear to all? Please let me know. You're finding something to eat. I can't even eat right now. You have the luxury to eat and study. Okay. All right. Moving ahead. So this is an important line here, that except for the plants in shade or in dense light is rarely a limiting factor in nature. Is that clear? In high, if the see, if we increase the intensity of light, then it can lead to breakdown of chlorophyll and decrease in the photosynthesis. Please let me know that all these points are clear to you. All these points are clear to you. Yes, please. All these points clear to you? Give me a thumbs up. Give me a thumbs up. We need thumbs up here. We need thumbs up here. We are almost going to complete our plant physiology here. Okay. Now coming on to the next factor, which is CO2. CO2. See here. Now, how does CO2 affect? Increase in concentration up to 0.05 of CO2 only can increase the rate of photosynthesis. Beyond 0.05, even CO2 will become toxic. Is that clear? So C3 and C4 will respond differently. And see here, both will show increase in the rate if we increase the CO2 concentration, provided light is increased. But C4 plants will show saturation at 360, while C3 will show saturation beyond 450. Why? Because C3 doesn't have CO2 enhancing or concentrating mechanism. C4 have their Krebs anatomy. They have that mechanism to enhance the CO2 concentration. So C4 will show saturation at a lower value, 360, while C3 will show saturation at a higher value. Is that clear? Now this is again important. C3 plants, for example, tomatoes and bell peppers, there are some greenhouse gases also, and they respond very well to the higher CO2 concentration. Are these points clear? Then C4 plants are well adapted to higher temperature, and C3 plants have a lower temperature optima. So with this, guys, we complete our plant physiology. Please let me know that these points are clear to you. These points are clear to you all. Please let me know that these points are clear to you all. All right, moving on to the next unit now, which is cell cycle and cell division. Cell cycle and cell division. See here quickly, very important chapter and some very important lines here. See, M phase starts with the nuclear division, which is karyokinesis, followed by the cytoplasm division known as cytokinesis. Is it clear? Now, in interphase, there are three phases, G1, S phase, and G2. G1 phase is metabolically active and cell grows in size. In S phase, three things are going to happen. DNA duplication, DNA will double, but the chromosome number will remain the same. Second, centriole duplication, and third, which is not written here but in genetics, histone proteins are also produced here. But majorly, other proteins are produced during G2. See here, proteins are synthesized during the G2, but not the histone proteins. Is that clear? Then, coming here, if the cell doesn't want to continue to divide and has to enter in a state of maturation or has to undergo differentiation, then cells which do not divide, they exit the G1 phase and enter into the inactive stage called quiescent stage, G0 phase. In G0, cells remain metabolically active but do not proliferate, do not divide. Now, M phase is the most dramatic phase. See here. In S and G2, new DNA molecules which are formed are not distinct and intertwined. Now, they start condensing and form chromosome during the prophase. In prophase, centrosome also moves towards the poles and radiates out those microtubules, and these microtubules are known as asters. Now, very important, the two asters and the spindle fibers together form the mitotic apparatus. Are these points clear? Now, at the end of prophase, GB, that is Golgi complex or Golgi body, ER, nucleolus, and nuclear envelope also vanish. They also disappear. In metaphase chromosomes, which are made up of two sister chromatids, they're held together by the centromere. Now, around the centromere are present small disc-shaped structures known as kinetochores. So spindle fibers are not directly attached to the centromere. Spindle fibers are basically attached to these small disc-shaped structures known as kinetochores. Is that clear? So they are the site for attachment of spindle fibers. Clear? It is characterized by all the chromosomes coming to life and one chromatid to each other connected to the kinetochore. Are these points clear to everyone? Please let me know. And then going on to anaphase. This is a PYQ. Centromeres split and chromatids, sister chromatids separate during which phase? Anaphase. What happens during telophase? Chromosomes have reached their respective poles. Now they will decondense and lose their individuality. So prophase and telophase are entirely opposite to each other. The nuclear envelope develops, nucleolus, GB and ER reform. Now when this nuclear division karyokinesis is done, it is followed by cytokinesis. See, in animal cell cytoplasm divides by furrow formation. So there is a invagination of the plasma membrane, that is furrow formation. While in plants, wall formation takes place. Now, wall formation starts with the formation of the cell plate. So it starts from center, grows outside, while furrow formation starts from outside, goes inwards, right? So this is a difference. So in plants, cell plate formation will be cell plate formation will occur, and this cell plate will represent the middle lamella. We know that there are three types of membranes in the cell wall: primary, secondary, and the common between the two is a middle lamella, right? So this middle lamella is represented by the cell plate. Are these points clear? Now, in case a plant shows karyokinesis, but it is not followed by the cytoplasm division, what will happen? What will happen? It will keep on increasing the number of nucleus. So, karyokinesis not followed by cytokinesis results in multinucleated condition known as syncytium. Example is liquid endosperm in coconut. Are these points clear to all? Please let me know. Quickly, please. Please, please, please. Are these points clear to all? So what is the significance of mitosis? We know that in social insects like bees, haploid cells divide by mitosis. We know that drones are haploid, right? Formed without fertilization. So their sperms will also be formed by mitosis. Is that clear? So otherwise, diploid cells formed, identical cells are formed by mitosis. Cell growth will occur because of mitosis, repair, healing because of mitosis. Now this is very important. Cell growth results in whenever the cell increases in size, it increases the protoplasm, it disturbs the nucleocytoplasmic ratio. Okay, so nucleus is the same, but the cytoplasm is increasing. So what is happening? The nucleocytoplasmic ratio is disturbed. In order to restore that nucleocytoplasmic ratio, cell divides. So what is that stimulus? What is that impulse for the cell to divide? It is a disturbance in the nucleocytoplasmic ratio, and it wants to restore that ratio. Are these points clear to all? Then coming on to meiosis, we basically see meiosis occurring in 2N cells while gamete formation, right? Now, meiosis has two sequential steps, meiosis 1 and meiosis 2, but there is single step of DNA duplication. Is that clear? So, one interphase followed by two phases. So, interphase, meiosis 1 and meiosis 2. Is this clear? So, there is one S phase, but two sequential steps of nuclear and cell division. Clear to all? Then meiosis involves a pairing of homologous chromosomes, and as a result of meiosis, four cells are produced, four daughter cells. While in mitosis, it was just two daughter cells. Now, this whole page is super duper important. I don't think that there is any page in your biology book which has so many questions coming from unlike this page. This one page is super special page. If I have to choose one page from biology book, it is this page, which has maximum number of PYQs. So we know meiosis is divided into prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 is the longest phase, divided into 5 phases, which is lazy, zebra, spondard, dare, disasters, leptotene, zygotene, pectine, diplotin, dikinesis. What happens during leptotene? Chromosomes become gradually visible under light microscope. You don't need an electron microscope here. Is this clear? Okay. Now see. So leptotene stage, compaction of chromosomes take place. And this is followed, and then comes the zygotine. So what happens during zygotine? Chromosomes start pairing. So paired homologous chromosomes, this pairing is known as synapses. So only homologous chromosomes will show pairing. So for bringing the homologous chromosomes together, there is a need for formation of synaptonemal complex. Is it clear? So now when the two homologous chromosomes, each with two chromatids, overall four chromatids lying side by side, we call it a tetrad. Or the two homologous chromosomes lying side by side, we call it a bivalent. Is that clear? So now these first two stages, leptotene and zygotine, these are short-lived. Followed by the third stage, which is pachytene. The characteristic feature of pachytene is crossing over. So here, what happens? The four chromatids, see, so there is crossing over. Crossing over means there is exchange of the genetic material between the two homologous chromosomes. Now, very important that it occurs between the non-sister chromatids. So this is a PYQ. These all lines are PYQs. I don't have to mark anything extra. Every line has appeared in PYQ. See, it occurs between non-sister chromatids of homologous chromosomes. So you have to watch the words carefully. Clear? And then this is an enzyme mediated process. Enzyme used during crossing over is recombinase because genetic recombination is taking place here, right? Then followed by diplotin. So now the dissolution of synaptonemal complex will occur. So this synaptonemal complex, which is bounded the two homologous chromosomes, it's going to dissolve and they are going to start moving apart. So homologous chromosomes separate from each other only except at a site of crossover. And now this X-shaped structure is known as chiasmata. So where is chiasmata visible? In the diplotin stage. So you always get questions in the form of match the following based on the characteristic features. Now understand, diplotin can last for months and years in case of some oocytes of vertebrates. Like in our case, it lasts for 11 years, right, from birth to puberty. Then the final stage of prophase 1 is dikinesis, marked by terminalization of chiasmata and total separation. Is that clear? Then metaphase 1, bivalent chromosomes align on equatorial plates, and unlike metaphase of mitosis, there are two equatorial plates formed here. In anaphase 1, homologous chromosomes are going to separate and not the sister chromatids. Please see this. Sister chromatids remain associated. Now, in telophase 1, again, opposite of prophase 1, but after telophase 1, that means before meiosis 2, no replication of DNA will take place. Meiosis 2 is exactly like mitosis. Only, see, the difference is in between difference between anaphase 1 and 2. Anaphase 1 shows separation of homologous chromosomes, while anaphase 2 shows separation of sister chromatids. Please let me know that these points are clear to you all. These points are clear to you all. Please let me know. So see, significance of meiosis helps in conserving the number of chromosomes in each species, increases the genetic variability, which is important for evolution. So with this, we complete cell division. With this, we complete cell division. Coming on to, I think guys, I have not downloaded the chapter of cell here. Let's start with biomolecules. I'll see for cell in a while. Coming on to biomolecules. Yes, please. Okay, let me see. I think I've forgotten to download that page. Never mind, we'll do that in the end. Okay, starting with this another dreadful topic, biomolecules. Let's make it easy. The important lines. How do we analyze the chemical composition of any tissue? So the okay. So the chemical used here, the solvent used here for analyzing the chemical composition is trichloroacetic acid. See the formula here. Okay. Now when we strain that tissue after dissolving, after making a paste of it in trichloroacetic acid, we get two components. One is filtrate, another is retentate. The filtrate is which is left behind, which is acid soluble pool, and of course which is left behind is the one which contains bigger molecules. And then we have retentate is the acid insoluble pool and has smaller molecules, bio micromolecules. Now this table is also important. You must know the At least the top 4 chemicals which are in abundance in Earth's crust, and then similarly in the human body. You rarely get question from match the following. Just leave it. Now, but you must know the increasing order of the top 4 or 5 elements in human body and in Earth's crust. Then, if we want to analyze the type of inorganic compounds present in the tissue, we go for destructive method. That means we burn the tissue, we make an ash of it, okay, and the ash will only contain the inorganic. See, the above method that we have used, in which the solvent trichloroacetic acid was being used, that method was to study the organic compounds, okay, while if we want to study the inorganic compounds in a tissue, we use this destructive method. Now let's talk about amino acids. Amino acids have alpha carbon because all 4 groups are different, so we also call them alpha amino acids. It is also known as substituted methane because all the 4 H in methane are replaced. So the 4 different compounds, substituent groups are hydrogen, carboxyl group, amino, and a variable group. So basically the difference between all the amino acids known to us is going to be through this variable group. Is this point clear? It is going to be through this variable group. Anyone with any doubt till now? Now see, the R group R group can be different. If R group is hydrogen, see here, if R is equal to H, then the amino acid is glycine. If instead of R we have methyl, then it is alanine. If R is the hydroxymethyl, it is the serine. Is this clear? So you can definitely get questions that in serine, what is the R group, and in alanine, it is a methyl group, and so on. Clear? Now on based on the R group again, and the number of amino and carboxyl groups, based on the number of amino and carboxyl groups, we have three types of amino acids: acidic, glutamic acid. Example of basic is lysine, then arginine, right? For neutral, we have valine, we have alanine, we have glycine. These are all neutral acids. For aromatic, means they have benzene ring in their R group. The examples of aromatic are tyrosine, phenylalanine, and tryptophan. Now, one very important and unique property of amino acids is that they form Zwitter ion. They form Zwitter ion. No other compound is going to form Zwitter ion. Why? Because they have 2 ionizable groups. One is the amino group and second is the carboxyl group. Both can be ionized based on the kind of solution they are in. So, at equilibrium, at simultaneously, this radical will have positive charge also and negative charge also. So such hybrid radical is known as Zwitter ion. Is this point clear? Now coming on to lipids. So lipids are made up of fatty acids and glycerol. Now based on the R group of the fatty acid and how many carbon atoms are present, see here, we have palmitic acid. Palmitic acid has 16 carbons, and now these 16 carbons include carboxyl carbon. It is called fatty acid because of the COOH group, right? So rest of the branch and then COH included, that palmitic has 16 carbons. You have to remember arachidonic has 20 carbons, again including carboxyl carbon. Is this point clear? Then fatty acids can be saturated, that means without double bond, and unsaturated with one or more double bond. Now simple example of simple lipid is glycerol. What is the chemical name for glycerol? Trihydroxypropane. Are these points clear? Now fatty acids are found esterified with glycerol. There are 3 types of fats: monoglyceride, diglyceride, and triglyceride, depending upon how many fatty acids are joined. See, there can be only one glycerol molecule, but there can be 1, 2, and 3 fatty acids. So one glycerol, one fatty acid mono, one glycerol, 2 fatty acids di, one glycerol, and 3 fatty acids is triglyceride. Is that clear? Now, what are oils? Oils are which remain liquid at the room temperature. Is that clear? Because they have lower melting point, they are always liquid. Is that clear? But if they become solid at the room temperature during winters, then we don't call them oils. Is that clear? Then we call them animal fats. Now there is another category of lipids which is phospholipids, which is found in the cell membrane. Maximum, they are found in the cell membrane. One example of the phospholipid is lecithin. Now what is a phospholipid? See this picture. sorry, this page. Let's come directly to lecithin. See, in lecithin is an example of phospholipid. We have phosphoric acid. Do you see this phosphoric acid here? This phosphoric acid makes this known as phospholipid. Lecithin is an important component of cell membrane, especially alveolar membrane. Is that clear? So this is palmitic acid. See, COH and then 14 here. 14, 15, total 16. So that is why C16 in palmitic acid. See the formula of glycerol. See these amino acids, R group. These in boxes are the R groups. Is that clear? We've already read. This is the structure of cholesterol. Now coming on to primary and secondary metabolites. Very important, you have to remember this table and under which category these compounds are coming in. So carotenoids, anthocyanins are pigments, morphine, codeine are the alkaloids. These are all in PYQs. This table is an important PYQ. Important PYQ. NTA loves this table. Okay. Then monoterpenes, diterpenes are the terpenoids. Grass oil, essential oil. Abrin and ricin is the toxin. Concanvalin is the lectin. Vinblastin and curcumin are the drugs. And these are the polymeric substances. Clear? Now, what is actually a secondary metabolite? Remember that. Secondary metabolites are those which are produced by organisms but not used by themselves. So they are not used by the host organism which is producing it. Is that clear? They're not used by the organism who produces it. It is used for the human welfare or they have ecological importance. Is that clear? Animals, as such, we don't produce any secondary metabolite. These are produced by, let's say, bacteria. See, it is written plants, fungi, microbial cells. They are known to produce the secondary metabolites. Are these points clear to all? Now, coming on to, coming on to the biomacromolecules. See, let's first talk about acid soluble pool. So acid soluble pool has biomicromolecules, right? Their molecule weight ranges between 18 to 800 daltons. Clear? But acid insoluble pool has macromolecules. Okay? They are proteins, nucleic acids, and phospholipids, but one micromolecule also, which is lipid. But lipid will be found in the acid insoluble pool. Is that clear? So lipid is actually a micro, but it likes to sit with the other macromolecules. Is that clear? In the acid insoluble pool. So all these macromolecules have their molecular weight except for lipid. Lipid has molecular weight around 18 to 800, right? But all the other macromolecules have their molecular weight in the range of 10,000 daltons or above. Is that clear? So why do lipids get along or come along with the other macromolecules even though it is a micromolecule? That's because we find these lipids in the cell membrane. So when we crush the tissue with the help of pestle in the mortar, so the membranes are broken and these lipids aggregate to form the vesicles. So we don't get an individual lipid as such. How we find lipids is in the form of vesicles, right? So these membrane fragments in the form of vesicles get separated along with the acid insoluble pool. Is that clear? So acid soluble pool basically represents the cytoplasmic composition. This is important. Now this table is again very important. So do we see the water is the most abundant component of the cytoplasm? Yes or no? Second is proteins. Third is nucleic acid. Fourth is carbohydrates, lipids, and ions. You must know this in the decreasing order as well as you should be ready for the match the following. See, match the following becomes very easy if you learn the decreasing order. You can easily go with the order, right? Are these points clear? Then again, this table, which tells us about the functions of different proteins, this table is very important. Collagen is intercellular ground substance. Trypsin is the enzyme. Insulin is the hormone. Antibodies fight against the infections. Receptors are for sensory reception like taste, smell, hormone, etc. And GLUT4, one very favorite for NTA. GLUT4 is glucose transport. This receptor, this protein helps in glucose transport into the cell. Is this clear? Now, these proteins are formed by the polymerization of amino acids. So, how many amino acids will participate in protein formation? Only 20. only 20. Now, since there are different amino acids different amino acids involved in the production, so we call protein as a heteropolymer and not a homopolymer. Are these points clear? Then collagen is the most abundant protein in the animal world, but in the overall biosphere, it is rubisco. Clear? Coming on to polysaccharides. Polysaccharides, again, cellulose is a homopolymer, starch, cellulose, starch, and glycogen. They are all formed from glucose. They are all polymers. Inulin is a polymer of fructose. Is that clear? Now, we get starch test. We do iodine test for starch. Why? Because iodine molecules can be held in the helical portion of starch. Cellulose will not form that complex helical structure, so we don't get any iodine test for cellulose. Are these points clear to all? Are these points clear to all? Now, moving on to the structure of protein, another very important aspect of this chapter. Primary structures. See, primary structure is represented by the line, linear arrangement. Left side is the first amino acid, right side represents the last amino acid. This was a PYQ. So, the first amino acid that is on the left hand side is the N terminal of the protein chain. The right side, which represents the last, is known as the C terminal. Is that clear? Now, in the secondary structure, this linear chain can go helical. Now, remember, we only have right-handed helices in our body. We only have right-handed helices of proteins in our body. Is that clear? Now, woolen ball-like structure is a 3D structure, also known as tertiary structure, very, very essential for biological activities. Then hemoglobin represents the quaternary structure, where hemoglobin has 4 subunits, 2 alpha, 2 beta, and they are all together. And every chain has, see, every unit, every alpha and beta unit has its own primary, secondary, and tertiary structure. Are these points clear? Now, coming on to enzymes, almost all enzymes are proteins. Okay? Not all. There are some nucleic acids which can behave like enzymes known as ribozymes. Are these points clear to all? Now, there are some enzymes which can even remain stable at very high temperatures and like temperatures up to 80 to 90 degrees Celsius. So such enzymes are isolated from thermophilic organisms like Taq polymerase. We know that. Is that clear? Now going on to the next part of the chapter. So this graph, again, very, very favorite to NTA. You can directly get this graph for labeling. So see this bigger graph, the upper graph, which shows activation energy when the enzyme is not used. And when the enzyme is used, the activation energy is lowered. See? And then on the y-axis, we are taking potential energy. On the x-axis, we are taking the progress of the reaction. Now, how do we conclude whether this reaction is exothermic reaction or an endothermic reaction? You see the gap here. If the energy of substrate is higher than the energy of products, it means it is an exothermic reaction. If products have lower energy, it means extra energy was all discarded out. So it makes it an exothermic reaction. So this graph 9.4 is an exothermic reaction or a spontaneous reaction. In case of endothermic reaction, energy of product will be more than the energy of the substrate. Is this point clear? Now, again, PYQ. What is the sequence in which this enzymatic reaction takes place? So, first of all, substrate binds to the enzyme at the active site, forming enzyme substrate complex, right? Now, when enzyme binds to the substrate, it induces the substrate. When the substrate binds to the enzyme, the substrate is going to induce the enzyme to change its shape. Is that clear? And get tightly fitted around the substrate. Then this active site in the close proximity will break the chemical bonds of the substrate and form the product. So now instead of substrate enzyme complex, it is the enzyme product complex. Then products will come out and enzyme will be separated again for reuse. Please let me know that these points are clear to you. So now guys, we have factors affecting the enzyme activity. First of all, we have temperature and pH. For both temperature and pH, we see this bell-shaped graph. That means If we take pH or temperature on the x-axis and enzyme activity on the y-axis, so we see that first pH will increase, as we increase the pH, enzyme activity will increase, and then after a certain point, the enzyme activity will start falling off. So that pH where enzyme activity is at the maximum is known as pH optimum. Similarly, we increase the temperature, the enzyme activity increases, but only to a certain extent. So the temperature at which enzyme shows maximum activity is T optimum, beyond which the activity again decreases. Then we have third factor, concentration of substrate. So we see initially as we increase the substrate concentration, velocity goes on increasing. But after a while, it becomes parallel to the x-axis. It does not increase further. Is that clear? So we get a rectangular hyperbola here. So maximum velocity obtained, if we see the corresponding half of the Vmax and the corresponding substrate concentration, we get Km. So what is Km? It is the Michaelis-Menten constant. It is the substrate concentration at which the velocity is half. Are these points clear to all? Are these points clear to all? Now, very important, inhibitor. Now, we have different types of inhibitors, but the inhibitor which resembles a substrate in its structure and inhibits the activity of enzyme is known as competitive inhibitor. Why? Because of the similar shape, it is going to compete for the same active site. Is this clear? Now, example of this is inhibition of succinic dehydrogenase by malonate. So malonate is the competitive inhibitor of succinic dehydrogenase. Another example is competitive inhibitors which are used for control of bacterial pathogens. So certain medicines that you take, certain like sulfa drugs, they are competitive inhibitors. Next, we come on to the classification on the base and see, this classification is based on the function that they perform. Okay, so oxidoreductases, you see, they will help in oxidation and reduction. You should be able to identify this equations given here. Also, you have to learn the name of these enzymes in a particular order. So the mnemonic is OTH lil. First. So these are 6 classes. See, it is clearly written. Enzymes are divided in 6 classes, and then each with 4 to 13 subclasses, right? Basically, every enzyme is given a number also. Clear? So oxidoreductase is class 1, transferase is class 2, hydrolase is class 3, lyases, isomerases, ligases, this is class 4, and so on. Okay, so if I tell you 4312, which class does it belong to? So the 4th class is lyases. Is that clear? So you should be able to decode that to which class that number of enzyme belongs to. Are these points clear to everyone? I will finish whole class 11. Yes. Now, another very, very important are the cofactors. So, these are the non-protein parts which bind with the protein part. See, there are certain enzymes which are only proteinaceous. We call them simple enzymes, while there are other enzymes which require a non-protein part. Now, these non-protein parts are known as cofactors. Clear? Now, protein part is called apoenzyme, non-protein part is known as cofactor. Very important. Now, if the non-protein part is organic in nature and tightly bound, it is called prosthetic group. If it is organic but loosely bound, it is known as coenzyme. If it is inorganic or a metal ion, then it is a metal ion. Okay. Now, example of prosthetic group, see here, is heme. Heme is a prosthetic group for peroxidase and catalase. So you can get this in match the following also. Then we have coenzymes. The example is NAD and NADP. Okay. Then we have metal ions. The example of metal ion acting as a cofactor is zinc ion for carboxypeptidase. Zinc ion for carboxypeptidase. Are these points clear to everyone? So it is so much essential for the enzymatic activity. If you remove the cofactor, the enzymatic activity will not happen. Is that clear? So cofactor and the apoenzyme in total forms the total enzyme known as holoenzyme. We call them holoenzymes. Please let me know that all these points are clear to you all. Yes, please. Please let me know that this was all clear to you. Moving on to the next chapter, guys. Next chapter. Wait. Okay. I am going to take morphology, anatomy, and structural organization tomorrow. Let's start with the first chapter here. Let's skip this. Morpho, anatomy, and structural organization is almost 12. So we have another big session. See, 12 has lesser number of chapters. So let's keep this unit 2 for tomorrow. Okay. I think we were over ambitious today. So let's skip this one chapter. This will be done tomorrow. Okay. Now coming on to living world. In living world, see, these are the important lines. Number of species that are known and described range between 1.7 to 1.8 million. So there is importance to standardize the name of living organisms, and this naming is known as nomenclature. Naming is only possible after we have correctly described correctly. Is that clear? And this is known as identification. Then full form of ICBN is International Code of Botanical Nature, and then International Code of Zoological Nomenclature. Then this idea of binomial nomenclature was given by Carlos Linnaeus. And what are the rules? There should be two names in every name. One is the generic name, the specific name. The names are in Latin. Okay? So first name is the generic name, second name is the epithet, second is the specific name. First is the generic, second is the specific name. Now when we are handwriting, they are separately underlined. When in prints, they are italicized. So this was a PYQ. So from this chapter, there are just two types of questions being asked. One is about the rules of the binomial nomenclature, and second is about the certain examples. Now see, genus name starts with a capital letter, specific name starts with a small letter. Then fifth rule is that the name of the author who described that organism and named it is written in the abbreviated form. So what is the correct way of writing the name of mango? It is Mangifera indica Linn. Clear? Then classification is all about grouping the organisms in convenient categories based on some easily observable characters. Is that clear? Then see, each category is known as taxa. Every category of different level is still called taxa. Process of classification is called taxonomy. All right. Now, modern taxonomy uses so many criteria: external, external structure, internal structure, structure of cell, developmental process, ecological information. They're all a part of modern taxonomic studies. Now, basic steps of taxonomy are characterization, identification, classification, and nomenclature. You have to remember it in sequence, CICN. So it happens in this way: characterization, identification, classification, and then nomenclature. Is it clear? Now, earliest classification, see, human has always been classifying things. Earlier classification was based on the usefulness of the organisms. Then the advanced form of taxonomy is systematics. In this branch of taxonomy or biology, we systematically arrange the organisms based on their evolutionary relationship. Okay, so systematics is one step ahead of taxonomy. In taxonomy, we don't consider the evolutionary aspect, but in systematics, we consider it. Again, the father of taxonomy and systematics is Carlos Linnaeus. Is that clear? Now, Linnaeus explained this everything in his publication known as Systema Naturae. Is that clear? So, this is systematics. Now, all the categories are coming under taxonomic hierarchy. So, category is known as unit of classification, termed as taxon. Clear? Category denotes the rank, and every taxon is a distinct unit of classification. Species is the lowest unit, and they are the organisms which have fundamental similarities and they have distinct morphological differences. Now, for example, Leo, Tigris, Pardus, they are all one species. They are different species, but slightly similar. So we keep them under one genus, Panthera. So under Solanum, we have two species, Nigrum and Melogena. So genus is a group of related organisms, like genus Solanum has potato as well as brinjal. Then Panthera has Leo, Pardus, and Tigris. Felis is all genus of cats. Then we have Petunia and Datura under family Solanaceae. So you have to learn these names as well. Okay, so these are important, but the most important taxonomic categories. This was a PYQ. Now this is the ascending order of the classification. Is that clear? As we go upwards, the number of similarities go on decreasing. And then this is the favorite table. And multiple times has this been asked, what is the scientific name of housefly, Musca domestica? To which family does it belong? Muscidae. So we don't know whether NTA still loves housefly only, or whether it has changed its new, or whether it's found its new love. So make sure that you learn this table. 1.1. Is that clear? With this, chapter 1 is also quickly revised. Then we have biological classification. We have biological classification. Okay, guys, let's quickly revise biological classification also. So, the first person to initiate this classification was Aristotle, earliest attempt. And what factors did he use? Simple morphological characters, and divided plants into trees, shrubs, herbs, and animals into one with RBCs, others without RBCs. But proper two kingdom classification was given by Linnaeus into Plantae and Animalia, but there was no distinction between eukaryotes, prokaryotes, unicellular, multicellular, photosynthetic, and heterotrophic. Is that clear? So basically, besides gross, two kingdom was used for a long time, right? So what were the criteria? Then came R.H. Whitaker. So this is another favorite table of NTA. So in this table, you can see there are 5 kingdoms. So one kingdom belongs to the prokaryotes, that is Monera, and 4 kingdoms belong to the eukaryotes. So we have already discussed this in a unit-wise revision also. So R.H. Whitaker, main criteria for his classification was cell structure, body organization, mode of reproduction, and phylogenetic relations. Is this clear? Then came the 3-domain system or the 6-kingdom classification. So in both these 6-kingdom classification, the kingdom Monera was further divided into Eubacteria and Archaebacteria. Is that clear? So 2 domains were given to Monera and 1 domain was left over for eukaryotes. Is that clear? Then, in earlier 2-kingdom classification, see, bacteria, blue-green algae, fungi, mosses, ferns, all were kept under plants, while, so according to that 2-kingdom classification, whether it is unicellular or multicellular algae, both were kept under one category, that was plants. Now, kingdom Protista, the 3-kingdom classification, when it happened, kingdom Protista basically brought out the unicellular organisms, both from plants and animals. See, Chlamydomonas and Chlorella were kept under plants, it was taken out and kept under and brought under kingdom Protista. And similarly, Paramecium and Amoeba, which were earlier placed under Animalia, was taken out and designated under kingdom Protista. Is this clear? So this was, so we have already discussed this chapter in unit-wise revision. Let's have a quick view of the important PYQs. See, bacteria live in extreme conditions. Thermoacidophils, halophiles, methanogens because of different cell wall structure, right? Methanogens in ruminants and help in methane gas production. So we have discussed this already. I've already given you pointers for these first 4 chapters in Unit 1 revision. So I'm just going to scroll through so that you can also check that you have underlined these important words and not missed on any. Is this clear? So this was all about the eubacteria. See, rigid cell wall, cyanobacteria, chlorophyll A, similar to green plants, flagellum, can be unicellular, colonial, filamentous, have gelatinous sheath, mucilaginous sheath. We remember we talked about it, cyanobacteria. Then specialized cells where nitrogen fixation takes place, known as heterocyst in nostoc and anabina. Then chemosynthetic bacteria, they produce ATP, right? They oxidize various inorganic substances to produce ATP. Recycling of nutrient takes place. Heterotrophic bacteria are the most abundant. So these are the uses we have already discussed. They are pathogenic also. They divide by fission and by production of spores. For mycoplasma, we remember 4 lines. Yes or no? Yes or no? So these are the things for Protista, not well defined. And this is one kingdom which links all other kingdoms, right? We have discussed that already. So diatoms and desmids in trisophites, freshwater or marine, float passively. So planktons, photosynthetic, 2 thin overlapping shells in a soapbox-like appearance. Silica walls in the cell wall. Silica in the cell wall makes it indestructible. Therefore, the remnants are known as diatomaceous earth, used for polishing and filtration, and they're the chief producers. Then for dinoflagellates, marine pigments, stiff cellulose plates, they have two flagella, one transverse and longitudinal. Then one example is red dinoflagellate known as goniolux and causes red tides, and it is toxic for the marine animals. Then euglenoids are freshwater, majority of them. They are not found in running water but in the stagnant water. Instead of cell wall, they have protein covering known as pellicle, which makes the body flexible. Two flagella, one short, one long. They're photosynthetic in the presence of sunlight and become heterotrophic when they are deprived of sunlight, and they predate, right? They have pigments exactly similar or identical to the higher plants. Then slime molds are saprophytic. So these are fungus-like protists. In suitable conditions, they form aggregation known as plasmodium. In unfavorable, they form fruiting bodies known which bears spores at their tips. And the spores, they have true cell walls, extremely resistant, can survive for many years, and when dispersed, they are dispersed only by the air currents. Then protozoans, they are all heterotrophs, predators, primitive relatives of animals, and based on their locomotory structures, they can be divided. Examples should be clear. Amoeboid is amoeba and entamoeba. Flagellated, which is trypanosoma, and it is parasitic, causing sleeping sickness. Ciliated, we have example of paramecium. And then sporozoans, which have spore-like stage. Example is malarial parasite or plasmodium. For fungi, we have white spots. We often see white spots on mustard leaves. This is a parasitic fungus. Which are we talking about? We are talking about albugo, right? Then there are some other disease causing fungi like rust causing puccinia. Then there are useful which can produce antibiotics like penicillium, grow in warm and humid climate. Unicellular fungus is yeast. Is that clear? Then long slender thread like structures known as hyphae. This is what the body looks like. And the hyphae form a network known as mycelium. Mycelium can be cenocytic hyphae, right? And if there is no septum, then it is cenocytic. If they have cross walls or septum, then it is not cenocytic. The cell wall contains chitin and polysaccharides. Then they can be saprophytes, parasites, and symbionts. Two examples of symbionts as lichens and mycorrhiza. Are these points clear? With reproduction occurs by three different means. So see, different type of spores. These are conidia, sporangiospores, and zoospores are the asexual spores, and sexual spores can be learned by boa. Basidiospores, ascospores, oospores, and we also have zygospores, by the way. Now, three steps which occur during the sexual reproduction. One is plasmogamy, followed by dikaryophase. So when the protoplasm fuses, but nuclei doesn't fuse. So N plus N. Followed by dikaryophase is karyogamy, that is fusion of nuclei. So karyogamy followed by zygote production, zygote formation, and then zygote undergoes meiosis, resulting in the formation of haploid spores. Please let me know this is clear. And guys, these steps of sexual reproduction occur in two classes of fungi, which is Ascomycetes and Basidiomycetes. Clear? Now, this is a very important line. This is a PYQ that the kingdom fungi is divided into classes on the basis of morphology of mycelium, spore formation, and fruiting bodies. And we have discussed all these lines in detail. I'm just scrolling through so that you can mark and check. We have recently completed our Unit 1 final revision, detailed revision. I hope we are thorough with all these lines. I hope we are thorough with all these lines. Yes or no? Please let me know in the chat that you have underlined these lines already and you are thorough. Yes. And give me a quick thumbs up. Yes, please. Give me a quick thumbs up here. All clear? All clear? So about plants, see, the insectivorous plants are partially heterotrophic. Bladderwort, for example, bladderwort, Venus flytrap, Cuscuta is a parasite. So plants show alternation of generation where diploid sporophytic phase will be alternated by haploid gametophytic phase. Please tell me. Animals store food is glycogen or fat. They show holozoic mode of nutrition and show definite growth pattern. Then viruses and prions and lichens and viroids, they were not categorized in five kingdom classification. Viruses are non-cellular. They are inert cellular structure outside the living cell. So Dimitri recognized it for the first time, discovered virus. Which one? TMV, which causes mosaic disease. Virus can easily pass through the bacteria-proof filters because they're even smaller in size. Bejer gave the term virus, called it venom or contagium vivum fluidum, infectious living fluid. Stanley crystallized them for the first time and said that it is majorly made up of proteins. Now, viruses have no other way of lifestyle but to be an obligate parasite. So, virus can either contain RNA or DNA, not both of them. Now, the genetic material is infectious. It is basically a nucleoprotein structure consisting of nucleic acid and the outer protein coat called capsid. Very important. Plants have single-stranded RNA containing viruses. So, plant viruses have, single-stranded RNA. Animal viruses have single-stranded or double-stranded RNA or double-stranded DNA, while bacteriophages have usually double-stranded DNA viruses. So the outer protein coat is known as capsid, made up of small units called capsomeres, which can be arranged in the helical manner or polyhedral manner. So these are the diseases which are caused by viruses in plants and animals. Is it clear? Then we have viroid, discovered by T.O. Dynia, smaller than virus because it doesn't have a capsid, so it is just free RNA and known to cause the disease potato spindle tobacco disease. Because it is just RNA, so it has a lower molecular weight. Coming on to prions, P for prions, it has just proteins, but these are abnormally folded proteins. Size would be similar to viruses, but it will not have any genetic material. Okay. So they are known to cause some very serious neurological diseases. For example, bovine spongiform encephalopathy (BSC), which is causing mad cow disease in cattle, and the similar variant which causes disease in humans is Creutzfeldt-Jacobs disease, or basically Crew is Creutzfeldt-Jakob disease. Now, lichens, as we know, is a symbiotic association of alga, phycobiont, and fungus, which is mycobiont. Algae is photosynthetic and fungus is heterotrophic. They come together. Fungus brings in the carbon dioxide and water, and algae having chlorophyll photosynthesizes the food. And lichens have ecological importance. They are very good pollution indicators, cannot grow in the polluted area. So guys, we'll stop here. So we are just left with unit number 2, basically, and Animal Kingdom. So we will be using tomorrow's time in completing these 3 chapters also from class 11. But I think big applause to all of us. We have been able to complete it in how much time? 7:30 we started. 8:30, 9:30, 10:30, 11:30, right? So it's just 4.5 hours and we have completed more than, and we are just short of 4 chapters, and we are done with class 11 syllabus. And now, why was I in so much hurry to complete it? Because tomorrow on guys, Sunday, 19th April, so 2 p.m. to 5 p.m. Tomorrow onwards, you have to sit for mock test series, free mock test series. The link is given in the chat box also. Click on the link and enroll for it right away. You cannot go to your NEET exam without sitting in these mock tests, because now that is a perfect recipe for failure, honestly, and for getting panic on the D day because you've never practiced it. So sit for the mock test series, get that real exam experience, get the live video solutions also for that. And then top 10 students, top 3 students, top 50 students will get mentorship through WhatsApp groups also. So there are different levels, but the whole or the main idea is you have to take that real exam experience before going for the real NEET exam. So take that test. So through every test you will be able to analyze your performance as well. So take that test tomorrow onwards, starting Sunday. Timings would be exactly same as your NEET exam, that is 2 p.m. to 5 p.m. So between 2 to 5 there would be no classes, hopefully, so that you can appear in the classes. Sorry, in the test. So join that. The link is in the chat box also. I'll see you tomorrow again, 7 p.m. We'll start a little early tomorrow. So we'll complete our class 12. Thank you so much for being with me from beginning to end. Let me know in the chat box, last chat, let me know how many of you have been with me from beginning till the end of the session. How many of you have been with me from the beginning to the end of the session? Wonderful. Thank you so much. All those of you who have attended the session. Let me know in the comment section. All of you have to tell that you have completed the entire session with me and you could revise everything. And now if you want to revise it for the second time or the third time, you can just increase the speed even more and revise it again. So we have hardly taken 4.5 hours, right? So if you increase the speed, you can have another revision of NCERT within, I think, 3 hours probably. So see, this is the use of such marathons. Thank you so much. Bye, everyone. Good night or good morning now. We have already started with Sunday. I'll see you Sunday again, evening at 7 p.m. with class 12 biology. Thank you so much and bye bye.